I'm really excited to open this day of celebration of basic translational and clinical research in the field of cancer. It's not a mistake. The World Cancer Day is indeed on the 4th of February, but we decided, since it falls on a weekend, we decided to meet and gather all of us here today in person and also to webcast live globally this amazing lineup of speakers. Even though we'll talk about the amazing progress that happened specifically in the last year and let alone the last decade in cancer research and approvals of new anti-cancer drugs, still we face 10 million people that die each year globally from cancer. There are 19.3 million new cases of cancer in a year. The estimated number of new cases of cancer in 2040 is estimated to be 30 million. And yet we know that 30 to 50% of all cancers are preventable. If we look at the cancer statistics, this is updated to January 2023, was just published. We still see that prostate cancer and breast cancer, to men and women respectively, are the leading cancers, type of cancers that we are facing with, followed by lung and colorectal cancers. And if we look at the estimated death, still lung is the leading cause for death from cancer, followed by, again, prostate and breast cancers for male and female respectively. Yet we saw that the FDA approved 10 new anti-cancer drugs in 2022, which represent 27% of all the new approvals in the last year. So imagine that from all the drugs that were approved from the FDA, almost a third were in the field of cancer. So that's quite impressive in terms of advancements and achievements. Yet we're talking about 10 new therapies, and this includes also technological approaches. And this leads us to the question why the numbers are still so low, and why we fail when we translate from the amazing breakthroughs that we see in the lab. When we take it to human clinical trials, we see that in the field of oncology, the likelihood of approval of a drug or of a compound that enters phase 1 is 5.3% to get an FDA approval. And one of the reasons that we and others, and including the FDA, thought that this can be improved is that the reason for this poor attrition rate is that the models are not representative enough to the clinical scenario. And about 2 weeks ago, the US Congress approved the Modernization Act 2.0, which means that maybe we should rethink the way we take compounds to clinical trials, forcing scientists and clinicians to first test our compounds on non-human primates and on rodents, at least 2 species of animals, as the numbers are so low when we look at the approval rate of 5%. And maybe we should consider human material on human on a chip, organ on a chip, tumor on a chip, and others. And this was signed and approved, as I said, just 2 weeks ago. So hopefully we'll see a better prediction of outcome in the clinic from those models. Time will tell. Hopefully in a year when we meet again in the 2024 World Cancer Day, we'll have what to tell you about it. So we have prepared an amazing lineup of speakers, but first, before we go to the talks, I would like to invite Professor Dan Pe'er, the VP R&D of Tel Aviv University, to welcome you.
Okay, thank you, Ronit. Indeed, cancer is still a leading cause of death, and I think that the discoveries that Ronit talked about which are amazing. Unfortunately, not all of them are translated well into the clinic, but in an optimistic standpoint, I hope we'll see much more in the next few years that will not only reach the clinic, but will be very successful. So I want to welcome all of you here and the people that are watching us. Indeed, there are here a line of really elite scientists from Israel, represent different fields in cancer, from basic to translational to clinical. And I want to wish you an extremely interesting day. Ask a lot of questions. And I think we're all going to learn a lot from this. Thank you again, and have a very successful day. Thanks.
Thank you, Danny, and I would like to thank Tel Aviv University, the Cancer Biology Research Center that are hosting this event, and the sponsorship of Teva Pharmaceuticals and Merck and the Biomed at Tau Hub for their continuous support of this event and in general of cancer research. So without further ado, I'm very happy to invite our first speaker, Professor Gidi Rechavi, the 2020 Israel Prize laureate, that will tell us about the advancements in cancer, both in basic research and in the clinic. Thank you, Gidi.
Good morning. It's a pleasure to be here, and thank you, Ronit, for putting together such a nice program in this lovely day. I'm going to discuss mainly cancer immunotherapy, and in this audience I don't have to elaborate more, that it seems that cancer immunotherapy is the major breakthrough in cancer research and cancer therapy in the last 15 years or so, and it seems that it's only the beginning. But I want to take you a few years back, actually 40 years, exactly 40 years back, to the work that I did when I was a graduate student at the Weizmann Institute with the late David Givol. This is Barbara McClintock, that looking at the maze, the colors of the maze, realized that this phenomenon is non-Mendelian, and she suggested and was awarded the Nobel Prize in '83 for suggesting the existence of what she called at the beginning a controlling element, what we later and today we call transposable genetic elements or movable elements, and the idea that are pieces of DNA and actually DNA or RNA that can move from one place to another in the human genome or in any genome and turn on and off genes. And it turned out to be a fascinating field. So she did it, as I mentioned, in maize. Later on, people showed that similar phenomena is found in bacteria, in yeast, in lower organisms. And then in 1982, we managed to identify the first example of transposition in higher organisms, in mammals actually. So, and also the first examples also showed us that such transposable element can take part in cancer. So this is the first paper we described exactly 40 years ago, where we found that a specific oncogene, at that time this was the beginning of the era of cellular oncogenes, so the CMOS oncogene was found to be rearranged in plasmacytomas, in the solid type of myeloma, and we cloned the wild type gene that is present in any normal cell and the gene that was rearranged. And we realized that at the 5' of the CMOS oncogene, we found insertion of novel sequences. And interestingly, when you take the wild type sequence and you cloned it and you put it in NIH33 cells, this was the routine assay for transformation, practically you see no transformation, no malignant foci. However, when you take the rearrange gene, it was very active in transforming fibroblasts to cancer cell. Then, so we published this paper and we were very happy. And then we were notified that the group of EdCAF at the NIH cloned and sequenced a transposable genetic element in mouse, which they termed intracisternal A particle, because there is the result of the expression of this retrovirus-like element was notified with this findings in electron microscopy, and we were asked by the editor of Nature, it's not so common that it happens, the editor called us and said, look, there is a sequence of EdCAF group, and the sequence that you published that was not known at that time, turned out that this is the same, the gene they identified is the sequence that was inserted in the CMOS gene, and we published another paper showing that it's exactly this intracisternal A particle, a LTR-like, a retrovirus-like element that was inserted. Then Mia Horvitz joined the team, and we were able to show that the insertion of the retrovirus-like transposable element inserted the long terminal repeat of the LTR of this element, actually inserted promoter and enhancer sequences which activate the CMOS oncogene. Then a few years later, we were lucky again. At that time, the only way to find such an event was luck. So we found another example, in this case, in a very strange tumor known as a transmissible venereal tumor that affects canines all over the world. And we were able to show that in this strange tumor that is transmitted by sexual intercourse between animals, there is insertion of another transposable element, in this time the line transposable element, upstream of the c-MYC oncogene. And interestingly enough, we studied tumors from all over the world, and all the tumors in the world have exactly the same insertion in the same nucleotide of the c-MYC, which proved that indeed this tumor was transmitted like an infectious agent. So it's transmission of cancer like infectious agent. Later on, there was a description of another such model, which is the Tasmanian devil. These animals are eliminated due to transmission of tumor that affect the airways of the animal. So this was the second example of activation of cellular oncogene by transposable genetic elements. And later on, we found more examples in mouse myelomas, and it was also shown to be relevant to human cancer. Then in 2001 came the first version of the human genome. It was realized that repetitive transposable genetic elements constitute almost half of the human genome. Just to remind you, the classical genes that encode for proteins comprise about 1.5% of our genome. Repetitive elements took over. These are a kind of genetic parasites that took over the human genome and the majority of other genomes. And so you see here the distribution. So we divide these elements to DNA transposons, which are very rare and usually not active in the human genome, but the majority are retrotransposon. They are transmitted via reverse transcription, and the most prevalent are the L1, the line element, the one that we found inserted into the MYC oncogene. This comprises about 17% of the human genome, and the LTR-like comprise about 8%. So, and there are more and more examples of such transposition. So Then came the technology that enabled you to sequence whole genomes. And 10 years ago, this was a science paper that analyzed about 100 tumors, human tumors, that showed that indeed such transposition are quite common in human cancer. And 2 years ago, there was a publication of about 3,000 cancer genomes representing almost 40 types of cancer, and turn out that transposition is very common, and there are tumors where it's more prevalent. For example, esophageal cancer is in esophageal cancer, in 95% of the cases, there are transposition events, and you see the red color indicates, so it's in about 20 or 25% of the cases, there are more than 100 transpositions per cancer, per tumor. So in some tumors, very, very prevalent. In others, it's less prevalent, but seem to be very, very relevant. And it has to do with activation of oncogene, inactivation of tumor suppressor genes, and other features. So in this slide, you can see all you need to know about transposable elements. So there are many versions of transposable element. They are short. The ALU sequences are only 300 base pairs long. The line element are 6,000 of them. They are not randomly distributed. There are elements that prefer to be inserted into gene regions. They are involved in mutagenesis. Insertion can inactivate genes or activate genes. They can do also other types of genomic rearrangement. For example, it's very common if you have two ALU sequences or two line elements. This can result in homologous recombination with deletion of large segments of the genome, inversions, and other events. It has to do with the control of activation. In the majority of cells, this huge number of repetitive elements are silenced mainly by epigenetic mechanisms such as methylation and histone modifications. However, in specific cells, for example, at the very early beginning of human development, in the very early embryo, and in germ cells, and interestingly, in the developing brain, such elements are allowed to jump. And as I mentioned, in cancer, it's very prevalent to see abnormal regulation and transposition. This insertion can affect both the germline and the somatic cells, and cancer is probably the best example. These elements are involved not only in transposition, but also in immune regulation, and I'm going to devote the majority of the talk to this element. They are also, underwent domestication, and many human and mouse and other species genes are derived, or at least pieces of the genes are derived from such repetitive elements. They are also involved in regulation. As I mentioned in the example that we found, if there is insertion of promoter, enhancer, or both in the vicinity of gene, this can change the control of expression, and thousands of genes in the human genome are regulated by such pieces of transposable element that were domesticated. And the last point is that until recently it was very problematic to study them. Remember, there are more than 1 million ALU sequences in the human genome. There are 400,000 of line elements. They are very similar to study them, to find very rare events of transposition. It's very tricky. Now with the new era of very long sequencing, it's becoming much easier. So coming back to immunotherapy, these are Ronjo and Allison that were awarded the Nobel Prize for the checkpoint inhibitors that you all know. And as you know, it's amazing advance in cancer therapy. However, only some tumors respond to checkpoint inhibitors. Some tumors are not affected until now. And even in those cases where there is a very significant response, for example, disseminated melanoma or metastatic lung cancer, still some patients will respond and others won't. And among the portion that do respond, over time, many of them will relapse. So we need to improve the efficiency of checkpoint inhibition, and a lot of groups are studying the factors that affect who is the tumor that will respond to checkpoint inhibitor and who won't. And we know that the load of mutations in tumor is relevant. The expression of PD-L1 is relevant. However, we learn more and more about the role of the microenvironment of the tumor. And we call it in a very primitive terms, they are the hot microenvironment that supports the immune response. In this case, you find usually a lot of cytotoxic and NK cells. The number and distribution of suppressor immune cells is lower. And in the cold microenvironment, where there is usually no response, it's the opposite. The number of active cytotoxic T cells is low. You have more regulatory cells, myeloid-derived suppressor cells, etc. And there is a major challenge how you can turn a cold microenvironment to hot microenvironment. And without going into all the details, it seems that the major factor in the type of microenvironment has to do with epigenetic control. And again, you know that for many years, we are aware of two major types of epigenetic control. One has to do with DNA methylation. The 5-methylcytosine has to do with the regulation of the expression of specific genes, but also has to do with the expression of repetitive elements and has to do with the control of the structure of the chromatin. It's very related to response to therapy. And the other major type of epigenetic regulation has to do with epigenetic regulation via marking of histones with a variety of chemical moieties such as phosphorylation, acetylation, ubiquitination, and many more. So over the years, and since 2015, there are more and more trials where people use drugs that can affect the epigenetic regulation of gene expression and chromatin in an effort to upregulate the repetitive elements, which, as I will show you soon, has to do with the control of the innate immune system. So here you see these repetitive elements, which, as I mentioned, in the majority of cells are silent by epigenetic regulation. When you use DNA methyltransferase inhibitors and acetylase and histone acetyl transferase inhibitors, etc., you can eliminate the silencing of the transposable element and upregulate the expression of such genes, such repetitive elements. And the RNA, and mainly double-stranded RNA, is recognized by cellular sensors. The best studied is MDA5, which activate the immune system, mainly resulting in upregulation of interferon expression and other NF-kappa B expression, other cytokines, etc. So until recently, these were the main efforts of upregulating the expression of transposable element to encourage the immune system. Our group is involved in the study of a new type of epigenetic regulation, which is called RNA epigenetics or epitranscriptomics. I mentioned modifications of the DNA, such as 5-methylcytosine. There are about, there are the 4 canonical letters of DNA and another 5 or 6 modifications that affect the DNA. When dealing with RNA, we are aware of 170 different modification nucleotides that are found in RNA. And until recently, it was impossible to study this modification in low-abundant RNA molecules. It was easier to study modifications that affect tRNA or ribosomal RNA that are found in large number of copies per cell. However, the rare RNA molecules such as messenger RNA, long non-coding RNA, microRNA, it was impossible. And our group actually opened this field when in 2004, the work of Erez Levanon, at that time a graduate student in the lab, now running his own lab at the Bar-Ilan University, we found the first modification, which is RNA editing. Adenosine is deaminated to inosine. And 10 years later, Dan Dominicini, again, at that time, graduate student, now a PI at the Sheba Medical Center. And this actually opened a new field. This is the epitranscriptomic field. And I'm going to mention in the last 4 minutes only the first modification that we managed to identify and study, which is the deamination of adenosine to inosine. And here you see, usually this is adenosine. It can be deaminated to inosine by enzymes known as the other enzymes. And if adenosine usually in RNA pairs with uridine, if you change adenosine to inosine, inosine is very similar to guanosine, then inosine pairs with cytidine. And importantly, this reaction can happen only in double-stranded RNA, only in those regions of the RNA which are folding back to result in double-stranded RNA. This is the substrate for the other enzymes. The majority of the first examples of the biological effect of editing has to do with recoding, change of one nucleotide from A to I. We change one amino acid. It has to do with recoding and change of the structure of protein, alternative splicing, etc. However, in our first paper where we describe the whole editome, all the editing sites in the human transcriptome, it turned out that the vast majority of editing events affect repetitive elements, and in particular, the ALU sequences, the short elements of the ALU families. And it's not surprising. On the average human gene, there are 17 ALU sequences distributed in introns, 5' and 3' UTRs. If there are two such elements in opposite orientation, they are very similar to each other, so this can result in a double-strand RNA. And remember, double-strand RNA is a substrate for other enzymes and editing. So it seems that in the vast majority of events, the role of RNA editing is destabilization of double-strand structure. So it was theoretically based on, we suggested it theoretically based on the database of editing sites that we found. And then Oz Solomon, another fantastic student, used a sophisticated methodology to map the structure of the RNA. And we were able to show, again, experimentally, that the main function of RNA editing is destabilization of perfect double-strand RNA structures. Why is this important? Because it seems that if we have hundreds of thousands of repetitive elements in the genome, they can result in double-strand structure. These structures activate the immune system. The cell recognizes a double-strand RNA structure as indication of a virus, and therefore we speak about viral mimicry by these elements, and the role of the editing system is destabilization of double-strand RNA structure and prevention of such events. There is a rare pediatric disease known as Aicardi-Gouttet syndrome, and one of the genes that has to do with this syndrome is ADAR1. Mutations in ADAR1 can result in disseminated inflammation, and this can be related to overactivation of the immune system. We were able to show that if you eliminate ADAR, for example, in liver cells, this results in inflammation. And we and others now realize that this is a tool. If we eliminate or downregulate ADAR, we can activate the inflammation in the tumor microenvironment. So if you don't have ADAR, the double-strand structure will remain active, will activate the immune system, and therefore this will result, this viral mimicry can turn the cold microenvironment to hot microenvironment. There are more and more indications that in this is working, and without going to many details, the use of silencing of ADAR can result in infiltration of the tumor microenvironment by activated cells of the immune system, and this synergizes with the checkpoint inhibitors and can improve the results of therapy. Just to mention, Yair Chizkiyahu, who is the student that is the main worker on this project. Itamar Goldstein and Dan Dominicini represent the immunology and the molecular biology with the help of many friends of the group.
Thank you, Gidi, for Thank you for taking us through the fascinating journey of gene editing and cancer. Any questions from the crowd? Yes, Michal.
Okay. The question is, can we control quantitatively, I believe, the level of activation? It's very similar to the way we activate the immune system by checkpoint inhibitor. In some patients, it's too little. In some patients, there'll be autoimmunity. So it's only the beginning. So far, it's only mice. So in mice, we can do it. But of course, turning towards human studies will need to be very controlled. And so right now, it's trial and error.
Maybe how do you envisage this being used in the clinic as prediction, as biomarker, or more?
Right now, there is a very nice paper published 3 years ago in Nature Communication, not by us, showing that there is a correlation between the level of expression of repetitive elements and the response to immunotherapy. So the biomarker is there. But of course, now we have to combine it with checkpoint inhibitors, like people are trying to combine more than one checkpoint, etc. So it can be a way to augment the response.
Thank you. So we have a really nice diversity of academic and clinical medical institutions in Israel. We're moving from Sheba Medical Center and Tel Aviv University now to the Technion, and I'm very happy to invite Dr. Keren Yitzhak to tell us about her research.
Hi, good morning, everyone. Thank you very much, Ronit, for the invitation to speak in this great event. And today I want to tell you about one of our recent projects. So my lab at the Technion, as Ronit mentioned, is a computational lab, which means that it tackles different challenges, computational challenges in cancer genomics. That being said, much of our efforts in the last 3 years have been focused on studying patient response to immunotherapy, and we are trying to tackle it from different angles, from the genomic, transcriptomic, and metabolic point of views. And today I'm going to focus on the genomic part. And our story begins with the set of somatic mutations that are accumulated in tumor cells, which everyone here knows that they are accumulated till the point that normal cells are transformed to malignant cells. And based on the set of somatic mutations, we can learn quite a lot of things. So first of all, we can identify the cancer driver genes, those genes that are mutated more than we expect by random, right? By studying somatic mutation in their 3 nucleotide context, we can also detect mutational signatures, like the UV signature, smoking signature, and apparently there are many, many more that are active in cancer genomes, and some of them are also predictive of response. And we also learned quite a lot about cancer heterogeneity, both across different tumors and also on intra-tumor heterogeneity. So how do we detect somatic mutations in cancer? This is a specific example of one of the many methods that were developed to do that. So the details are less important, but the basic concept is always the same. And this is that we need tumor samples, but we also need a normal sample, a matched normal sample from the same individual in order to distinguish between the somatic and germline mutations. And in a previous work, we asked ourselves a simple questions. What if we just replace the DNA from the tumor with RNA from the tumor? Supposedly a very trivial task. The RNA is a copy of the DNA, so this should have been pretty easy. Apparently it was a bit more complicated because we tested that and we took tumor samples that had both DNA and RNA co-extracted from the same sample. We applied this method for identifying a somatic mutation called MUTACT, and we just compared the mutation list that we got, okay? And this is the picture that we found. In blue is the number of mutations we found in the RNA. In red is the number of mutations that we found in the DNA. And I'm sure you can see that there's a problem here. That apparently, there are many, many more mutations in the RNA, but we quite immediately figured out that this is noise that we somehow need to filter out. But specifically, this is how our data looked like. So obviously there is an overlap, right? There are mutations that we see both in the DNA and in the RNA, but there are mutations that we see only in the DNA. We don't find them in the RNA. And this is the first question that we asked ourselves, why is that? And the other probably bigger problem was to filter out all the noise that we saw only in the RNA. So in order to address the first part of why there are mutations that we see only in the DNA but they are not in the RNA, we did a simple power calculation. Again, without getting too much into the details, what you can see in this plot is that only the yellow part have sufficient statistical power, okay, to identify these mutations in the RNA. But most of the mutation, we simply don't have enough statistical power to detect them in the RNA. And the reason for that is mainly because they are not expressed, okay, or not sufficiently expressed. So if the gene is not there, we cannot detect the mutations in it. There are more reasons like allele-specific expression and others, but they were actually the minority of the cases. And in order to address the second part of this problem of filtering out all the excessive mutations that we see in the RNA, we developed a pipeline called RNA MUTACT. Again, I will not get into the details, but it has a bunch of filters that are specific to RNA, noise that is specific to RNA, sequencing noise, I mean, alignment problems and things like that. And eventually, after applying it, it was able to filter out 98% of the false positive, okay? And it barely had an effect on the true positive, okay? On the ones that were also in the DNA. And then also quantifying it in terms of sensitivity and specificity, our results were quite good. So a question that you may have asked yourself in the last 5 minutes or so is why, right? Why does you want to do this? Like, finding mutations in the RNA, not in the DNA. What's the big deal? So the first answer to this question is that you would like to do this when you don't have DNA, right? And specifically, what motivated us to apply this method and develop it is that we wanted to learn about the accumulation of somatic mutations in normal tissues, okay? This is what we wanted to do, but we wanted to do that on all the tissues in our body, on hundreds of individuals, and simply there is not such data. Okay, there are no DNA from such big cohorts, but lucky us, there is a project that did exactly that, but only with a small difference that they sequenced the RNA, not the DNA, because their goal was to study expression in normal tissues. So again, this is not the focus of this talk, but we found quite a few mutations, somatic mutations, and also in cancer genes that are accumulated in normal cells. There are tissues that accumulate more, tissues that accumulate less. And if someone wants to learn more about it, it's already published, and I'm also happy to talk about it in the break. But the second potential answer to the question of why using RNA, not DNA, is the cases where RNA is the more relevant metric. And what do I mean by that? So I mentioned at the beginning that we are using the set of somatic mutations to identify driver genes, signatures, tumor heterogeneity, and more things. But in the last few years, and as Gidi mentioned, we have immunotherapy, and the set of somatic mutation has also been used in order to estimate the tumor mutational burden, which is basically defined as the number of non-silent mutations that are found in the tumor genome. And indeed, in June 2020, the FDA actually approves the use of anti-PD-1, pembrolizumab, for all solid tumors with high tumor mutational burden. So in general, the process is such that mutations are accumulated in the DNA, transcribed to the RNA, generates mutated peptides, right, that are presented on the cell surface by MHC class I molecules and elicit an immune response. But as I just told you, we found that most of these mutations are actually not found in the RNA, like around 70%, which means that they will not generate mutated peptides and will not be presented to the immune system. So our thought was, okay, let's try to estimate the tumor mutational burden directly from the RNA and not from the DNA, and perhaps it will give better results because the clinical trial based on which the TMB was approved as a biomarker of response actually had only 29 overall response rates, patients with the high TMB. And we know also today in the clinic that this is far from being the perfect marker. So there is a room for improvement. The problem that we tackled when we wanted to do that is that most of the RNA sequencing that was done from patients does not include the matched normal sample, okay? Because the goal was to study gene expression. This is what people are usually do when they sequence the RNA. So our challenge actually, and what I wanted to tell you today, is about a method that we developed for detecting somatic mutations directly from the RNA, but without the matched normal sample, okay? And this boils down to the following challenge of distinguishing between the very large number of germline mutations that are marked here in orange and the relatively small number of somatic mutations. So we started collecting features about these mutations. These were read-level features and mutation-level features, and we looked on a lot of normal samples and on a lot of databases that contain germline information. And we started exploring these features, and we actually found that most of them significantly differentiate between somatic and germline variants. However, when we tried to use each of them and find the optimal threshold that will separate these two groups, the performance was pretty bad, okay? You can see that in the graph over here, mostly the precision was very low. And this motivated us to use a more sophisticated method related to machine learning that can perhaps combine these features into a more predictive model. So this is exactly what we did. We developed a new method that starts with data that is labeled, which means that we have the matched normal sample, so we know which are the germline and which are the somatic mutations. We collect all these features. We train a model, okay, that learn these features. Specifically, we used random forest and applied five-fold cross-validation. And then, using these models, we now take samples that have only the RNA. We detect all the mutations, which are now mixed, either somatic or germline, and we apply these models in order to classify these mutations. And now the only question that remains is how accurate are we in doing that? So here is the performance of our method. We first applied it to melanoma, where you can see that both our precision and recall are around 85%. And there's also very high correlation between the number and the set of mutations found in the RNA when we use the matched normal versus our pipeline, which does not require this sample. We also applied it to another two cancer types, for lung adenocarcinoma and for colon cancer. In lung, we actually got even better performance. In colon, the performance was a bit worse, mainly because it is a less mutated cancer, okay? So while the number of errors that we have did not change, okay, entire set of somatic mutations that are accumulated in colon sample, it just captures a larger fraction. So overall, the performance is lower. And now we basically wanted to see if we can use the set of somatic mutations that was predicted by our model to identify driver genes and mutational signatures, right? These are the type of analysis, the follow-up analysis that usually people do using using the set of somatic mutations. So first, for driver genes, we collected a set of known melanoma driver genes, and we wanted to see if we can correctly predict mutation in these genes. You can see that we were actually got a very good result for this set, but also, and more importantly, we could also identify driver genes de novo. Okay, so we just put in the set of somatic mutations, and we apply this algorithm called MUTIC to identify driver genes. And in this plot, you can see that out of the 24 driver genes that it identified, 22 of them were also predicted by our model. And we also were able to find mutational signatures that were identified both in the DNA and in the RNA, again, without getting too much into the details. And because I have only 3 minutes left, I'll move on to the main result, which we were finally ready to estimate our TMB metric, which is derived from RNA. And we first applied it to melanoma patients that were treatment naive, and actually for melanoma, it was found that patient with very high TMB, a very low survival. So we excluded these patients. You can see them in the survival plot. They are marked in green, and you can see that they have very poor survival. But then when we split the patient to those with high and low tumor mutational burden, we could see that the expressed TMB, those that is derived from the RNA, had better performance than the one that is derived from the TMB, from the DNA. And finally, we applied it to data set for patients that were actually treated with checkpoint blockade. We took only the RNA was available. We took the RNA, we applied our pipeline, and we got the set of predicted somatic mutations. We first validated them. Okay, we saw that we can identify the driver genes correctly, the mutational signature, etc. And actually, what was interesting about this data is that these were pre-treatment patients. Some of them were treatment naive, and some have previously progressed on anti-CTLA-4. And in the treatment naive, there was no association with survival, either in the DNA or in the RNA. But for those that have previously progressed on EP, it looked like the authors originally reported there is no association with survival. But when we used our new metric of expressed TMB, we actually saw that there is a significant difference between these two groups. So to summarize, we developed a new method that can detect somatic mutations directly from the RNA. It can now be applied to all RNA-seq that is out there, and it can also be used to compute the TMB. Of course, there are still things that we need to approve and improve. So first, most of the samples are FFPE samples outside, and we need to see that our pipeline works on them too. And it also did not slip our mind that there is a significant improvement with the expressed TMB, but it's not huge. Okay, so there is much more work to be done. So we are now trying to reconstruct such cancer-specific models. So we can apply this to different cancer type and study the set of expressed mutations and their predictive power. But also we are working on building integrative predictors that can take also factors from the tumor microenvironment and other genetic factors into account, hopefully generating better models in the future. So credit here goes to Autumn Katsir, a wonderful postdoc in the lab, and Noam Rudbeck that actually took this and made this into a tool that everyone can run very easily. And of course to my wonderful group, funding agency, and thank you for your attention.
Thank you, Karen. That was really fascinating. Any questions, Asaf?
This is exactly the type of things that are found in our, sorry, I will need to repeat your question. So Asaf is asking about correlation of this with neoantigens, no neoantigens, and so on and so forth. So it's on our endless to-do list. It's there, and of course it's very important, but we haven't done that yet. This is exactly the integrative predictors that. Yes, Moshe is asking if we try to add it to factors that are active in the stroma and the immune system. So this is the kind of integrative predictors that we are trying to build right now, taking all these factors into account, and maybe it will generate better models.
Do you isolate the tumor cell compartment from the environment, or you?
This is what
on a bulk?
it's bulk. Yeah, we have also worked in single cell, but specifically to make it more clinically applicable, we are trying to do everything from bulk. And to separate the stroma and immune, we use deconvolution methods, but it's not that easy.
Of course, nothing is that easy. A question? If not, thanks again to Karen. So back to Tel Aviv University. Our next speaker is Dr. Asaf Madi.
Thank you. So, good morning, and thank you for coming, and thank you for the invitation. It's really exciting to be here. So our lab is systems immunology. We do a lot of mouse tumor models, single-cell RNA, immunotherapies, bioinformatics. But today I would like to share with you something a little bit different. It's a collaboration with the lab of Ronit Sachi-Fainero. And as I mentioned before, we do a lot of mouse tumor models. But mouse tumor models have many limitations. And one of the main limitations is the complexity of the tumor tissue. Okay, in human cancers, you can see here examples from breast cancers, from melanoma, from kidney cancer. You have these immune niches. These are niches where that looks or resembles a T-cell zone of secondary lymphatic tissues. And they are made up of dense areas of antigen-presenting cells, APCs, and stem-like CD8 T cells. And these are super important because these CD8 stem-like cells are the ones that give you the proliferative burst following checkpoint blockade. And they are also generally correlated with better prognosis. So to further investigate such complexity, we need better in vitro models. However, the current 2D models are lacking this type of complexity. So what do we have? On one hand, we have these 2D models, as mentioned before. They are cells grown on this rigid plastic. And on the other hand, we can work with in vivo models or with tissue samples. And in between, we have these 3D models, spheroids and organoids, which are great, except that they are usually self-assembled. And as such, they lack the necessary complexity to understand such complex interaction or special organization. Okay, so at the end of the day, if we want something like this, a tissue with immune niches inside of it, we need to generate in vitro something like that, right? We need some tumor cells that are embedded with immune niches. And with this in mind, we thought, okay, it's going to be very simple. All we need to do is take some tumor cells, some dendritic cells, some CD8 cells, use a 3D bioprinter, and generate this type of a structure. And this work was led by Lena Neufeld from Ronit's lab and Sarah from my lab, and is currently in progress also with Anchika and Opal. And it's quite simple. So you take this 3D bioprinter and you try to generate the type of design that you want to generate. And we've been working on it for the last 3 to 4 years. And what have we been doing is exactly what I would like to share with you, except for generating this small but delicious sandwich. Okay. So, before we started, we needed to initiate some basic immune and non-immune properties, because these type of properties are the ones that are going to dictate the type of questions that we are going to ask and the type of experiments that we are going to do. So the first one was, can we really generate very complex structures using a 3D bioprinter? And the answer is yes. This is exactly what this technology was designed for. But more importantly, can this construct really survive for longer period of times? And our initial experiment shows that you can keep these structures for 2 weeks and even longer, and the cells are just fine. Second thing, can we use microscopy, fluorescent microscopy, confocal microscopy? No problem, obviously, once we color the cells. The third one is quite complicated. We wanted to see if we can estimate tumor killing. Okay, and we can do that by estimating cell viability by elements that are looking at the metabolic rate of the cells, cell viability. The fourth one is, can we measure secreted factors from these structures from the media? And yes, we can take liquid from the media and use ELISA to measure this type of elements. And interestingly, you can see here that when we printed tumor cells together with CD8 T cells, we get less of this metabolic rate, meaning more tumor killing. And when we print and more of this secretion of TNF alpha as expected from the T cells. The last thing is, can we isolate the cells from these structures that we've generated? Because this is very important for downstream analysis. For example, from FACS that you are seeing here, or other more sophisticated analysis like single-cell RNA-seq, etc. And yes, we can isolate the cells from these structures, and not only that, we can make sure that we do it without destroying the cell surface molecules, which are very important for follow-up experiments. Okay, now that we've established some of the basics, we can move on to more sophisticated properties. The first one is, can we add factors to these structures and induce properties of the cells inside the structures? And yes, we can do it with cytokines, obviously, because these structures are getting their nutrients from the media. But here you can see an example where we used a larger molecule, antibody checkpoint blockade, anti-PD-1, and you can see here increase in TNF alpha following the adding of the anti-PD-1. And the last thing is, can we isolate specific parts from these structures? And yes, we can do it. We can use scalpels if the structure is larger, and we can use a puncher-like device that is used for biopsies. And obviously, we can also use more sophisticated methods like laser capture microdissection and isolate specific regions of interest from these structures that we are generating. The next question that was interesting to us is, can we learn something about T-cell infiltration? So we have this structure that we've generated, and now we are putting some T-cells on top of this structure, and we want to see if these T-cells can infiltrate into the tumor, into the structure, and kill the tumor cells. And cutting a long story short, yes, we were able to do this type of assay. And not only that, what was interesting is that we saw that we have increased infiltration of these T cells when they are forming this type of aggregates on top of the surface of the tumor structure. And this is super interesting. And it's sometimes, it feels like a playground, and playground is great because it really increased the creativity of the students that are doing this type of experiments. And this is a very preliminary results. In fact, everything that I showed you is preliminary results, but these are super preliminary results. We shows you the tumor structures. So the tumor cells are here in blue, and then you have these immune niches from dendritic cells and CD8 cells in these organized structures. And it's very similar to what you saw before with the actual human tissue samples, except that this is super organized. This is a OCD kind of a tumor. So again, we can track the movement or killing of the cells using microscopy. We can analyze secreted factors by ELISA. We can apply external factor, even checkpoint blockade, and we can isolate cells for further in-depth analysis. But as I mentioned before, the importance of these immune niches have been shown before, but it was only correlation, okay? People have found that these immune niches are correlated with better prognosis, with better response to checkpoint blockade. But the basic question remains, would we see a difference if we take the same number of cells and put them as a mix or organize them into immune niches? And this is exactly what we did. And again, these are super preliminary results, but what we see is that, yes, it matters. So when you have these immune niches, you have more of these tumor killing. And I think this is super great. I'm super excited about these results. Okay, so now we thought that, okay, we can generate these lymph-like structures inside the tumor, but what about the real thing? What about a real tumor-draining lymph node? So I looked at Google, and this is what I found. So you have the cortex, the paracortex, the medulla, the capsule, and clearly this is not this is too much for us to do with a bioprinter. So again, I googled tumor draining lymph node plus cartoon, and this is what came up, and this is something that we can work with. And again, this work was led by Lena from Ronit Sachi-Fanero's lab and Karen from our lab. And I didn't mention it before, but before we go into the bioprinter, we need to generate some kind of a design. So this is the design of the tumor draining lymph node, and I know it's a super simple kind of a design, but this is what we like to start working with. Once you have this design, you feed it into the bioprinter, and then you can generate a model using some kind of pluronic gel with some food coloring, and this is the end result of this model. And once you have the model, you can move on and work with live cells. And it's hard to see, but you can imagine that here you have this tumor draining lymph node from live cells inside the well. And these are some really beautiful EVOS fluorescent microscopy where we use different colors to differentiate between combinations of different cell types. So in the germinal centers, you have the B cells, the dendritic cells, the macrophages. In the paracortex, you have the T cells, and this is the macrophages. And the capsules, you have the DC. So really a lot of combinations of different cells, and we can do many more. And now if we zoom in using a confocal microscopy, we can see this even in a more clear kind of a picture. So in red you have the DCs, the macrophages are dark blue, and then in green you have these T cells. And you can see here that some regions are not perfect because generally the technology is not perfect, but it's definitely something that we can work with. And here is something that I really wanted to emphasize, is that we can really focus on these regions that are in between, for example, the germinal center and the cortex, or any other type of specific spatial interactions. So this is super cool, at least I think so. But the next question was, okay, we have this tumor draining lymph node. Can we flow some lymphatic fluid through it? So our first design was not sufficient for this type of understanding. So we needed to generate something more sophisticated, and this is what we came up with. So the entire structure of live cells is inside this structure that is surrounded by non-live cells, and this would allow the flow of fluid, let's say lymphatic fluids, through this device. And following this type of design, we generated the actual live cells. And you can see here that yes, we can flow some fluid through this device, and it comes in contact with all the cells that we are intended to do so. And then once we have that, we can connect it to a more controlled kind of a flow, and put this entire thing into an incubator, so we can perform the experiments over longer periods of time. And to summarize what I've showed you today, so I showed you that yes, we are able to generate very complex structures which are geometrically defined. This is super important. I showed you the immune niches and the tumor draining lymph node. I didn't mention the reproducibility, but yes, we can generate many of those structures. And immunological properties, this is what we care about, and hopefully you're going to see more and more results around these areas. We can track the movement of cells using microscopy, killing of cells using metabolic rate kind of indicators, analyze secreted factors, simple ELISA, apply external factors, including checkpoint blockade, as I mentioned before, and isolate cells for further in-depth analysis, functional assays, single-cell RNA-seq, whatever. And with this, I'd like to end and thank the people that were involved in the work. So from my lab, Sarah, Karen, and Ayelet. From Ronit's lab, it's Lena, Anshika, and Opal. And of course, my collaborator. Without them, this would not be possible. So my main collaborator, obviously, is Ronit Sachi-Fainero, but I did get help also in mice and some reagents from Shlomit, Stefan, and the late Nir Friedman, and this was supported in part by the Morris Kahn Foundation. And thank you for listening, and this is our great team.
Thank you, Asaf. Great work. Yes, Gideon.
Okay, so physiology, this is a big question, and we're taking it one step at a time. First of all, what I showed you, that they can respond and they do respond. But the question, do the B cells actually are forming some kind of a functional germinal center? We didn't prove it yet. This is on our to-do list. But as mentioned, this is one step at a time.
In general, I can comment that the whole technology is now being validated in a clinical trial of 80 patients in Sheba Medical Center. So we're looking at the response of the patients to immunotherapy or chemotherapy, and what we see in our models from that patient with hers PBMCs flowing in those systems. So we'll know. Moshe?
Yeah. So how long can we keep the cells in these devices? So it all depends on a few parameters, mainly the composition of the matrigel that we are using. And surprisingly, it's much longer than you can keep them in a 2D culture. Okay, so they survive longer. In these early experiments, we can keep them for at least 3 weeks, and they are fine.
Any other questions? No, thank you, Asaf.
Thank you.
Now moving to Bar Ilan University, our next speaker is Professor Mira Bardasad. Thank you.
So I would like to thank Ronit and all the organizers for inviting me. And today I will walk you through targeting intracellular checkpoint molecules from the mechanism to potential novel anti-cancer strategy. So I will actually focus on 2 projects. The first one, we target the actin nucleation promoting factor, the Wiskott-Aldrich syndrome protein. And actually the big advantage of this protein that it's exclusively expressed in hematopoietic cells in order to find a potential approach for hematopoietic malignancies. And the second project is that we target intracellular inhibitory checkpoint of natural killer cells in order to enhance their anti-tumor activity. And this was done by nanoparticle. The first project was done by small molecule compounds. So for the first project, we actually focused for the last several years on a protein called the Wiskott-Aldrich syndrome protein. And this protein is exclusively expressed in hematopoietic cells, and it's actually a key regulator of actin cytoskeleton. And that's the reason this protein is actually a regulator of very important cellular activities such as cellular activation, proliferation, motility, and tissue invasiveness. All these activities that I just described are actually the hallmark of cancer. So this protein is actually in autoinhibitory conformation in naive cells. However, once they are being activated, small g GTPase, such as CDC42 in a GTP form, associate to the GTPase binding domain of the protein, releasing its autoinhibition, and so that the VCA domain of this protein is now can associate to a complex of protein called ARP23, which increased the nucleation of actin filaments. So we actually found that this protein wasp undergo ubiquitination, which leads to its degradation. And we asked ourselves, where is actually the degradation site? And we found out that it's located on the WH1 domain, on the N-terminus, and specifically on lysine 76 and 81. When we mutated this lysine to arginine, we actually blocked the degradation of the protein. So this was our suggested model, that in naive cells, actually WASP in its autoinhibitory conformation, and there is a protein called WIP, Wiskott interacting protein, that associate to the WH1 domain of WASP and protect WASP from degradation. And upon cellular activation, we found out that there is a partial dissociation between WIP to WASP, so that the degradation site is exposed for the E3 ubiquitin ligase, the c-Cbl and CBLB, so they ubiquitinate WASP and induce its degradation. So we ask ourselves, how can we take this basic knowledge to potential therapeutic approach? Can we induce the natural degradation of WASP in hematopoietic malignancies? And by this way, block actually WASP-dependent activities such as cellular activation, proliferation, motility, and tissue invasiveness. So we actually started by actually using artificial intelligence in order to identify small molecule compounds that can interact to the WH1 domain of WASP, to the N-terminus, actually causing to dissociation of WIP from WASP, and as a consequence leading to the natural degradation of this protein. Obviously, once we found these few SMCs, small molecule compounds, we had to verify that indeed they associate experimentally to WASP and induce its degradation, affecting WASP expression level, affecting WASP-dependent activity, and also to prove it in vivo, in a mouse model. So I'm not going to take you through the in silico workflow. It was published, but I will say that we actually screen hundreds of thousands of small molecule compounds from drug-like based libraries. We found few that associate to the WH1 domain of WASP. And by the way, this part of the work, the in silico screening, was done in collaboration with Professor Yanai Ofran. And these few small molecule compounds, we could actually check them by microscale thermophoresis, to see whether they indeed associate to WASP. And you can see that there is a very nice binding between one of the SMC13, to WASP, about 30, the KD is about 30 nanomolar. This SMC did not bind WASP in the absence of the WH1 domain, neither WASP homologs that express in non-hematopoietic cells, such as NWASP or WAVE2. When we submit this work for publication, the reviewers came back and said, you know, you have to show it in an alternative approach. So we used the BLR, the biolayer interferometry, to show that indeed SMC13 associate in a specific manner to WASP, and in the absence of the WH1 domain, there is no binding to WASP homologs. When we treat the cells with MG132, which is proteasome inhibitor, we get a ubiquitinated WASP, a smear of ubiquitinated WASP, and we get back the protein. In contrary, cells that were not treated with this proteasome inhibitor. Moreover, actually, we produce some cell lines that exclusively express either WASP wild type tagged to WIFP or WASP without the WH1 domain tagged to WIFP, and we show that these cells that actually express WASP in the absence of the WH1 domain, they're actually in the presence of SMC13, there is no degradation of WASP, in contrary to cells that express wild-type WASP. Later on, we wanted to show actually the association or the interaction between WIP and WASP. So we used FRET, fluorescence resonance energy transfer technology. We tagged WASP with CFP, we tagged WIP with WIFP, and if the emission spectra of the CFP goes to the excitation of the WIFP, we should get FRET, and this is something we could measure. And indeed, you can see that the FRET efficiency between WIP to WASP is actually about 37%. However, in the presence of this SMC13, the FRET efficiency actually dramatically reduced. We moved to the in vivo model, And we used SCID NRG mice. We grafted them with human non-Hodgkin lymphoma cells. Once the tumor reached 100 millimeter at the third, we injected this small molecule compound, IP. We measured the weight of the mice, and then we extracted the tumor and checked for WASP expression level as well as for WASP-dependent activities. So you can see a huge difference between the negative control group treated with the vehicle only versus those treated with the SMC13. There is a huge difference in the tumor size, and you can see it also here in the images. When we actually extracted the tumor and checked for WASP expression level, we got a reduction of about 64%. And when we checked proliferation of the cells, there was a reduction of about 75%. And we also measure the migration of the cells. And these two movies, but by the way, were taken in the same setting. And you can see that there is a huge difference. Those treated, mice treated with SMC13, their cell were inhibitors by about 75%. So to summarize it, SMC13 might serve as a potential hit for medicinal chemistry to be used as a potential approach for hematopoietic malignancies. So I would like now, with your permission, to switch gears and to move to the other project. And this project, if in the previous project we actually focused on the cancerous cells, blood cancerous cells, in this project we mainly focused on natural killer cells. And we actually target intracellular inhibitory checkpoint in order to increase NK cell anti-tumor activity. So we all know that natural killer cells play a major role in the immune defense against viral infection and tumor growth, and they do that by secreting cytolytic granules that kill the target cells and by secreting cytokines that serve as a bridge between the innate and acquired immune system. And you can see here this movie, natural killer cells, transparent, attack the cancerous cells, which are labeled with green fluorescent protein, and within minutes, the cancerous cells are being killed. So for years, we actually investigated the NK cell signaling cascades. And we know that NK cells possess both activating receptor over their surface that goes to the activation of several adaptor molecular enzymes that eventually lead to cytotoxicity, to the release of cytolytic granules and the killing of the cancerous cells. But natural killer cells also possess inhibitory receptors that are connected in their cytoplasmic tail to a phosphatase called SHIP1, which dephosphorylate this activating signaling protein, blocking ERK MAP kinase activity, blocking cytotoxicity. We actually found that we identified additional substrate of SHIP1 in natural killer cells, and we found that PLC gamma, a very important enzyme that is responsible for calcium signaling, is one of the substrate of SHIP1, is being dephosphorylated, and actually also another substrate cannot, by dephosphorylating via SHIP1, cannot actually recruit PLC gamma to the cell membrane, and actually this inhibit calcium release, inhibit the release of calcium from the ER, and inhibit NK cell activation. So SHIP and also CBL. CBL is an E3 ubiquitin ligase that ubiquitinate the activating signaling proteins such as LAT, translocating them to the proteasome for degradation. These two are key regulator of NK cell activation. They are actually an intracellular inhibitory checkpoint of NK cell activation. So we ask ourselves, how can we take this general knowledge into potential applicative approach? So we prepare nanoparticles. These nanoparticles are liposomes, and they are coated with anti-NKp46 antibody that specifically recognize natural killer cells. And they encapsulate siRNA to CBLs and SHIP, and they specifically integrate into natural killer cells and actually dramatically enhance their activity, anti-tumor activity. These nanoparticles are labeled with, are fluorescently labeled, and the big advantage of this approach versus the common used one is that we are actually target intracellular signaling molecules that are downstream to several inhibitory surface receptors. With the inhibitory checkpoint blockage, you actually target a specific receptor or two receptors. Here, you actually target a key, a junction of an intracellular inhibitory checkpoint. And so it's much more effective. In this approach, we don't have to isolate the natural killer cells from the blood of the patient. We are not facing any cellular contamination or insufficient NK cell number or a reduction in that cytotoxicity. You can see that the nanoparticles do not affect the volume of the cells, neither the density of the cells. They specifically incorporate into natural killer cells, those that express NKp46. Cells that do not express NKp46, such as erythroleukemia, do not incorporate the nanoparticle, as you can see here in red. And also the gene silence Sibyl sheep to about between 60 to 80%. So we try to figure out what is actually the role of this nanoparticle and their effect. So we actually measure intracellular calcium flux. And you can see that cells that were treated with nanoparticles encapsulating Sibyl and sheep, their calcium flux was dramatically increased versus those treated with non-specific siRNA. So their activation was much higher. Also, we check NK cell degranulation, coenzyme B secretion, and this is from the NK cell side, and also from the cancerous cell side, we check target cell lysis, and we find a dramatic increase in these activities. When we took it to the in vivo model and took mice, KidNRG mice that were engrafted with human non-Hodgkin lymphoma cells, and we injected once the tumor reached 100 millimeter the third, we injected human natural killer cells, and we also, a day after, we injected the nanoparticles, and then we extract the tumor and check for the natural killer cells and characterize them. And you can see that actually the tumor the mice that were treated with the nanoparticles encapsulating the Sibyl and sheep siRNA, their tumor growth was abolished versus those treated with non-specific siRNA, and moreover, actually, except of the big difference in the tumor size, the natural killer cells that were isolated from this tumor were much more vicious killers. So this was just recently published. It was the cover of the EMBO Molecular Medicine journal. And you can see here nanoparticles in yellow integrate into natural killer cells in pink, manipulate their cell behavior. They become serial killers towards cancerous cells in purple, which raise white flag. I would like to thank all the people that led this research, to Guy Bieber, Aviad Ben Shmuel, and Abhishek Putinvitil that led the first project, to Batel Savag and Guy Biebers and others who led the second project, to our collaborators, Anayo Fran, and also from the Sheba Medical Center, Professor Avigdor Abraham and Dr. Mirav Kedmi, and thank you all for listening.
Thank you, Mira. Great translation to the clinic now. Hopefully, we'll move forward. Any questions from the crowd?
So they are actually coated with anti-NKp46 antibody. Thank you for your question, because I have to thank Professor Angel Pogodor that gave us this antibody. And they are very specific to natural killer cells. NKp46 recognize natural killer cells and not other cells. And we proved that. I mean, in the paper, there is much more data that I couldn't put in there. Short talk.
Of course,
Okay.
my question is regarding the nanoparticles. Which kind of nanoparticles are there? There are pegylated liposomal that are,
Yes,
the
they,
peg is labeled with the antibody?
yeah, they are actually liposomes, and there are several layers. It's actually self-made nanoparticles. We made them in my lab, and it was like we construct them, over time and try to pick up the best one that will be useful for this system.
Thank you. Again?
Thank you.
Last but not least, from before the break, we move to Ben Gurion University of the Negev, Professor Moshe Elkabetz.
Thank you very much, Ronit, for the invitation, and thank you for making it accessible for the taxpayers that funds parts of the research that we show here. So I would also like to thank my previous speaker that gave the introduction for immunotherapy. And the basic aim of immunotherapy is just to enhance the interaction and the interaction with the tumor cells or with dendritic cells that eventually will kill better the tumor cells. But this interaction is quite complex. It regulates by plenty of different ligand and receptors that determine the efficacy of the treatments. And among this long list of potentially therapeutics targets, so far the PD-1 and PD-L1 show the most robust activity in patients. And over the years, there are more and more different cancer types that have been approved for treatment with anti-PD-1. But what you also can see that among this long list of tumor types, there is still very little knowledge about the biomarker of response. And that would lead to the knowledge so far that although that many patients are actually eligible for this treatment, relatively low number of patients are actually responsive to this therapy. And since the interaction or the activity of the antibody that blocks anti-PD-L1 or PD-1 regulates the interaction between the two proteins, it was very makes sense to look for the abundance of the PD-L1. And among the different biomarkers that we heard before, PD-L1 stain is one of the marker. But I'll just give you an example about the challenges that we face in the clinic, and this is just an example of staining of PD-L1 in lung cancer, and you can see that the pathologists need to sit under the microscope and evaluate the intensity of the slide, and they give a score between 1 to 49 or above 49, and that could be associated and known to be associated for some cancer type with the response. But we also know that even patients that have low level of PD-L1, they still do response. And what are the challenges? That we have diversity of staining between, even in the same tumor. You can see here different antibodies that indicates for different staining level. And more than that, when pathologist sits under the microscope, he needs to define where the PD-L1 is expressed, and there are different scoring for different cancer types. So you can see that it's TPS, CPS, and it really depends on the cancer type. And this is an example of a recent clinical trial that used PD-L1 staining. You can see here, this is the differences between PD-L1, the curve in blue, compared to the standard of care. And you can see that obviously there is an additive value of using immunotherapy in this case. And here you can see the curves when you have high CPS. So obviously you need the magnifier to see the differences, because the additive value is around 10%. It's a lot of people, it's very important, but obviously it's not a game changer. And here come the hypothesis of our research, and we say, okay, abundance and expression is important, but maybe what is also important is the interaction between the ligand and the receptor. And if we will be able to develop a system that can quantify the functionality of the ligand, maybe it will give a better indication. So for that, it's a joint project with Professor Angel Progador, and we are together working on it for several years already. And I'm glad to have the MD PhD student Bar Kaufman that took this challenge, actually been able to take it from a vision into action. So what we have done, we hypothesize that if we will develop a biosensor that can quantify the functionality of the ligand, and it will be sensitive enough, then maybe it will be can be used to predict the response in cancer patients. So what we have done, we engineered T cells from mouse fused with the human PD-L1 together with a zeta inside the cells. So once the receptor is activated either by a ligand or by an antibody coated to the plate, the receptor can be phosphorylated intracellular, and that will give us a signal inside the cells, and the cells will secrete cytokine IL-2, and that will be our readout for activity. So basically, we can mimic it as the tumor microenvironment. So this is our biosensor. We call it ICAR. And once it interacts with the ligand, it will secrete IL-2. And once we block this interaction, or there is no ligand, functional ligand, there will be no IL-2. So this is the principle of the entire system. So what you can see here we coded different amount of PD-L1 and recombinant PD-L1 and PD-L2 into the plate, and we seed those ICAR cells in the plate, and you can see that the response of the ICAR to PD-L1 and PD-L2. And what you can see here, that the PD-L2 actually can activate the ICAR better than the PD-L1. This is actually associated with what we know in the literature about the affinity of PD-L1, of PD-L2 to PD-1 that is greater than the PD-L1. So the question is whether we can now take it into one step forward into measuring the functionality of ligands with cell lines. So what we have done, we took multiple tumor cell lines and we quantified the surface expression of PD-L1 and PD-L2 using flow cytometry, and then we did a co-culture of the ICAR cells together with the tumor cells to let them interact with each other, and once they interact, they will secrete the IL-2. And then we could also block either PD-L1, PD-L2, or PD-1, and to quantify the total activity of the ligands or the contribution of each one of them. In addition to that, we also took the tumor cell and we induced the PD-L1 and PD-L2 expression by activate them with the cytokine. We use interferon gamma and TNF alpha, it doesn't really matter, but what we saw that we have more expression, and now we can also look on not only in the basal level, but also in the activate level. So let's see how the data looks like. So here you can see the expression of PD-L1 and PD-L2. This is a flow cytometry base. So basically, if it goes to the right, means that you have greater expression. So in the baseline level, obviously, there is relatively low level of PD-L1 in this specific example. So you can see that after the induction, you have an upregulation. And this is also true for the PD-L1, for the PD-L2. And here is the co-culture and the secretion of IL-2. So you can see that in the baseline, obviously you have low amount of IL-2 because we have only a little bit of the PD-L1. But following cytokine activation, we have an induction of PD-L1 and PD-L2, and that was associated with very high stimulation of IL-2. And when we block the PD-1, obviously we block it. And when we block either of PD-L1 or PD-L2, we can contribute the contribution of each of the ligand functionality. And since for PD-1 it's very simple, we have only two ligands, you can see that the total of PD-L1 and PD-L2, the sum of PD-L1 and PD-L2 is actually the total of all. So when we look on the data of multiple cell lines, you can see that there is diversity in the baseline expression, but also you can see that there is upregulation in the activity of PD-L1 and PD-L2 upon cytokine secretion. And we took all the data and we could see that we have a very strong association between the activity and the surface bound of the PD-L1 and PD-L2. And since we have two, we can also see that once we can measure one and we can estimate the other one. And this is basically what you can see here in the data. So now we want to take it another one step forward, and to try to make it more helpful for the society, we thought that it will be important to try to adopt this system, not to measure the ligands in a fresh, which is quite challenging, else to be able to quantify the functionality in a FFPE tissue, which is much more accessible for us. So for that, we took the models that we have generated over the years in the lab. So we use a patient-derived xenograft, meaning that we took a tumor from patient, implant them into the mouse, and now we have as isogenic models of PDXs and FFP from the same tumor. And the whole motivation here is to be able to find the optimal condition that we will be able to look for the functionality in a fresh and FFP. This is what I show you here, it's a summary of 1 year of work of Bar Kaufman. So what eventually the process is that we take the samples, we have been generated a unique of antigen retrieval, and then we see the antigen retrieval after we constitute the functionality of the ligand on a plate. We adhere it, and now once it's adhered to the plate, we can add the live ICAR cells, let them interact with the tissue, and therefore we can later measure the IL-2. And this is the data, how it looks like when we compare fresh to FFPE. And obviously you can see a very strong correlation between them. However, you can also appreciate that we lose a little bit of the signal with the FFPE, meaning that we still have some room for improvement. Since it's not so trivial to quantify it on a FFP, we need to take into consideration multiple parameters, and you can see here just an example of how the tissue looks on the plate. We need to make some normalization, including the amount of tissue that in the plate. So for that, what we have done, we make a score. So from now on, I will tell score of PD1, which indicates for the total activity of the PDL1 and PDL2, score for PDL1 indicating for the functionality of PDL1, and score for PDL2 is the functionality of PDL2. And what you can see here that eventually the data of the all 5 PDXs, you can see that the 3 of the PDXs, 1, 2, and 3, the major activity of the ligands actually comes from the PDL1. In the PDX4, it's mainly PDL2, and in PDX5, we had both PDL1 and PDL2. So after we established the system in a preclinical model, we ask, okay, whether we can actually test our hypothesis in cancer patients. So for that, we work together with our great collaborator from Soroka Medical Center and Barzilai Center, and we collected 42 patients in this cohort, among them 19 responders and 23 non-responders. You can see here the distribution of the patient. The majority of them are lung cancer, some renal carcinoma, TCC, few skins, and a couple of head and neck cancer. So when you look at the cohort, you compare responder and non-responder. Basically, there is no bias between the two groups, no differences in stage, no differences in sex. And later we had some information about the clinical information of those patients. So you can see here the staining of the PDL1, as I showed you, as I introduced you in the beginning of the talk. And what you can see that among the patients that they have PDL1 expression, higher 50% and 49%, you can see that actually it's what we know, that the majority of them will respond. However, the ones that have low responder, most of them do not respond. But we can see that at least a few of them did respond. So when you look on this calculation, it basically gives this type of sensitivity score, and you can see that the AUC it's around 0.5. In our cohort, it's a little bit lower than what it's on the literature, that is around 0.65. So now I'll show you the data of our ICAR score. So what you can see here is in the left bar is the responders versus the non-responders, and here we can see the score of PD1. I will remind you that the score of the PD1 is the total functionality of PDL1 and PDL2. And what you can see that among the responders, we have some patients that have higher level of PDL1 and PDL2 functionality. But we also can appreciate that among the non-responders, all of them had quite low amount of functional PDL1 and PDL2. And when we look on the different response, complete response, partial response, stable disease, and progression disease, you can appreciate that we have significant differences in PD-1 score between the complete response, partial response, compared to the non-responders. However, we still don't have a sufficient amount of samples to distinguish between stable disease and progression disease. This is one of the things that we are working on it right now. When you look at the overall survival, you can see, sorry, I forgot to mention that we did a cutoff of 4, which is the standard deviation of the mean afterwards. And what you can see that when we split the cohorts between patients that they had higher ICAR score higher than 4 and lower score, we've been able to clearly differentiate between the responders and non-responders. And what you can see that the patient that they have low PD-L1 score, unfortunately, they did not respond well and passed away much faster compared to the patient that had a high PD-L1. And here you can see the AUC, that it's relatively high, and we need to confirm it, obviously, in additional clinical setting. So we took the challenge now to also look, not the challenge, the opportunity to look for the correlation between staining and the scoring. And what you can see here, this is the 18 patient that we have both data of the staining and the activation. And what you can see that there is no really association between both. And we try to understand, to get a little bit understanding why is that. And what you can see here, those are the patients that were stained low for the PD-L1, but they had high level of ICAR score. So when we look on the data, we saw that actually in those patients, they had relatively high amount of PD-L1 activity. So meaning that although that the expression is low, still the PD-L1 is active, very active, and they have also PD-L2. And then the vice versa, that we had some patient that they stained very strongly for the PD-L1, but the activity was low, and we saw that in those patients, potentially the PD-L1 is not mature enough to induce the suppression of the T cell. So let's summarize. I showed you the work that my group together with Angel Progador developed, the ICAR system that is mimicking the functionality of PD-1, and using this system we'll be able to measure the functionality of PD-L1 and PD-L2, the two ligands. We have been able to use the checkpoint blockers to block PD-L1, PD-L2, or the PD-1 to get the quantification of each functionality of each of the ligands. And we have been able to adjust it to the FFPE, and using that we can do retrospective studies, and that's what we have done with 2 cohorts. Today I showed you 1, but we have an additional work, and we saw so far a good correlation between response and the activity of the ligands. And I would like to thank my group, but the student that led this, that's still leading actually this project, is Bar Kaufman and Angel Progador, and obviously the funding. Thank you very much.
For this interesting highlight differentiation between the PD-1 and PD-L2. Questions, Asaf?
So the question, if I've been able to look for a spatial, like how it's expressed on the tissue. We are still not in the phase to get into resolution of localization, but this is something that we always have in mind, how we can basically fix the cells, the ICAR cells in the position, and then later align it with the functionality. This is the to-do list. Yes, so the question is whether the PD-L1 that expressed on the CD8s can influence the 80. Okay, so the 80 is for the CTLA4, right? So the ICAR that we develop is not dependent in any other immunocheckpoint. So it means that we we design it in a way that we can measure what is exists, but it cannot be inhibited by other receptors, I would say, or ligands. So, but it's referring to your question, is it's not interfering, but I think in the vision-wise, we should look on the multiplexing system to measure not only the functionality of one ligand, else to use it, to adopt it in a way that we can measure the complexity of the heterogeneity. And this is, again, in the to-do list, which will take a while to make it actually functional. So the question is whether PDL, the interferon gamma or the cytokine can measure the functionality. So for this specific, I would assume that you refer to the fact that we stimulated the cells, the tumor cells with the interferon gamma to upregulate the ligand. So the answer is no. And because we have done those experiments also when we stimulated the cells with interferon gamma, we fixed the cells, removed the interferon gamma and measured the activity. So it does not affect, and this is the way that we balance it.
Thank you, Moshe.
Thank you very much.
So we'll take a half an hour break now. We'll reconvene at 11:30. Thank you to all the wonderful speakers of the first session, and we're looking forward to the second one. So welcome back, everyone. Happy to continue this fascinating morning with excellent speakers, and now moving to Hadassah Cancer Research Institute in Jerusalem and the Hebrew University with Michal Lotem, Professor Michal Lotem. Thank you for coming.
So Ronit, everybody opened their talk saying, thank you for letting me speak. So I want to say thank you for letting me listen, because it's such an amazing opportunity to do this. The topic of my talk is alternative splicing of immune receptors, a fast intramolecular regulatory mechanism with therapeutic implications. And usually when we talk about alternative splicing, we speak of this as a source of protein diversity. And briefly, it's the process that would skip out or take out introns, bind exons one to the other, and different configuration can be to the final resulting mature messenger RNA that can include or miss some of the exons of the original gene, and therefore different proteins will emerge as a result of the different exon inclusion in the transcript. But what actually I want to describe in the context of immunology, immunity, and anti-cancer immunity in particular today is that alternative splicing is much more than a source for protein diversity, but from studying physiology of lymphocytes, what we can see is first that alternative splicing in the context of immunity is a very fast, rapid intramolecular regulation. It kicks in faster than the process of transcription, and the outcomes of alternative splicing in immune cells can be either the generation of a protein which is a dominant positive reactor or a dominant negative reactor out of the same gene. It could be the generation of a decoy element, or it could result in a molecular poison that shifts out the code and the sequencing. So I am an oncologist, and alternative splicing in the context of cancer is discussed a lot in cancer. And just to compare this to alternative splicing in immunity, in cancer, usually we speak about aberrant splicing, glyc mutations in splicing factors. In the context of immunity, we speak about that as a physiological splicing process, and the primary trigger for the splicing is the T-cell receptor activation. In cancer, this is a source of uncontrolled proliferation. In immunity, it is a source of fast regulation. In cancer, alternative splicing usually disrupt regulatory mechanisms, and in immunity, it's an intrinsic balance of regulatory receptors. Nowadays, there's a lot of interest in alternative splicing in cancer as a source for neoantigens, and what I want to show you is that the physiological alternative splicing in immune cells can be a source for new strategies for immunotherapy. So in order to see the dynamics of what happens to alternative splicing in T cells following activation, what we actually did or received material from a big experiment taking naive CD4 and later CD8 T cells, activating the T cells at time point 0 and harvesting RNA a long time, and then performing RNA sequencing using lengths of at least 100 nucleotide in a paired end method. Also, we took original or derived from other paper RNA sequencing from patients receiving immune checkpoints for metastatic melanoma, and the patients were distinguished to responders, non-responders. The T cells were intratumoral lymphocytes. And lastly, the methodology to do the splicing detection, the alternative splicing variant ratios out of a same gene were based on RMATS software, and in this software, special weight is given to sequences which, if present, report that a certain junction between exon and exon exists in the transcript, and if missing, this means that a certain junction between exons is not existing, and so it can tell then that an exon is missing or skipped out. And a lot of the informatics that I will show was done by Elad Zisman, an MD PhD program student that I mentor in collaboration with Dr. Keren Avraham from the Technion. And our assumption in the beginning when we set to analyze which splicing isoforms, variants, appear in different time points in lymphocytes along activation, we actually searched for these types of patterns that will also be associated with a general increase in the transcript. So we're looking at splicing and transcription that are going up together. And so, not surprisingly, among the top hits that we received were several immune-related genes, such as CTLA-4. You see that CTLA-4 shows up twice because it means two different splice isoform switches or changes along activation, and you can see FAS, you can see interleukin-2 receptor alpha, etc. But then when we disconnected splicing events from the amount of the general transcript of an RNA, what was very interesting, and this, the CD4 T cells actually shared with the CD8 T cells, is that doing enriched pathway analysis for the spliced genes, the genes that are differentially spliced along activation, we found that the major pathways are actually the ones relating to messenger RNA splicing or to other processing, like processing of capped intron containing pre-mRNA, ribosomal RNA. So what seems to be is that the reactive alternative splicing occurring following T-cell activation in first place affected genes that regulate splicing. If you ask which are the genes that we can see top expression, not splicing, then of course it is immune-related genes like early rising genes, CXCR4, intermediate rising genes, interleukin-7 receptor, other chemokine receptors, and late risers. Among them you can see interleukin receptor alpha, etc. So trying to discriminate to which or what amount of similarity or clustering can we see in genes related to expression, we can see that at the early time points, you don't see genes of expression very much diversing from one time point to the other. The difference in the clustering starts much later on. It starts in 24 hours, 72 hours, then the transcript picture of the lymphocytes really changed dramatically. When you look at splicing, it is just the opposite. So the splicing at 3 hours are very much dissimilar to 6 hours or to 12 hours. Then the general splicing pictures gets stabilized in a much later time points, like 24 hours, 72 hours, etc. So what are the correlates of what we had? I call it the roadmap in the tube of activating T cells along time. What are the correlates to real life, to what you can see in patients? So the data is driven from RNA sequencing, RNA sequencing of tumor infiltrating lymphocytes taken out of melanoma patient at Massachusetts General Hospital, and sequencing the cells before the patient start treatment receiving a PD-1 inhibitor-based therapy, and the patients were discriminated between non-responders to responders. But now we are looking at the time point before treatment was given to the patients. And what you can see clearly is that while in the non-responding patients, and this is the non-responding patients in this correlation heat map, you can see that actually they show nothing, or they resonate nothing of the T-cell roadmap of splicing events, those that later will not respond to the treatment. While responding patients already before they received immune checkpoint inhibitors, you can already identify in their T cells patterns of splicing that are typical to at least early time points of activated T cells. This series was published by Sam Freeman and others. When you look at the splicing patterns in non-responding patients but on treatment, they start to get more similar to the T-cell splicing roadmap. The patients that were later defined as responders are even more similar. Their tumor-infiltrating lymphocytes, splicing selections or favorites were much more similar to what you can see in the tube in activated T cells. Just to show you an example, this is a sashimi plot, a way that we describe or demonstrate splicing events, and so that each arc demonstrate a junction, and you can also see the level of the transcript. You can see, for example, FIN, a serine threonine kinase active in T cells. You can see that in responding patients, this exon is much more frequent to be transcribed, while in non-responding, this exon is skipped, while the other exon gets priority. To summarize this part, the majority of splicing events take part in messenger RNA splicing. A splicing pattern typical to activated CD4 T cells is found in patients likely to be immune checkpoint inhibitor responders, and following treatment, both responders and non-responders will display splicing preferences of activation. Can this be exploited in any way to treatment? So we in the lab use two methodologies to affect or to interfere with splicing. One methodology is to use splice switching oligonucleotides, and the other is to do splice disruption using CRISPR edits. So the idea behind the splice switching oligonucleotides is that you design and do some chemical modification on a sequence of about 15 to 20 nucleotides that will complement to an area where the spliceosome would need to recognize in order to do the cutting and to start the process of skipping out introns. The way to do this using splice disruption is to insert a mutation at a splice site this way again to make the splice site non-recognizable. First, we try to do this in PD1, and doing this to PD1 gene, what is the picture for this gene? You can see that in non-activated naive CD4 T cells, PD1 is actually not apparent at all. 3 hours following activation, you see here 3 exons, and what I will tell you is that this exon number 3 on the gene, actually encodes for the transmembrane segment for PD1. And you can see, roughly speaking, that in about 1/3 of the transcripts, this exon is excluded. In about 2/3 of the transcript, the exon is skipped out. If the exon encoding for the transmembrane domain of PD1 is actually skipped out, what will be generated is a protein that will be secreted, and this protein can be a decoy for PDL1. Later on, you can see already at 6 hours post-activation, much more so, 24 and 72 hours, there's no more skipping of the transmembrane domain of PD1. All PD1 that will be transcribed will be membrane bound. And so Shai Tzaban, in my lab, an MD PhD student, actually did a process that we call oligo-walk that tests successive oligonucleotides to mask the splice junction for exon 3. She found several splice switching oligonucleotides that you can see when she's testing them on T lymphocytes. You can see that the normal transcript for PD1 now is changed, and the full-length PD1 is decreasing, while that part missing the transmembrane segment is now increasing. And when she evaluated T cells, and this is just part of several features of T cell activation and function that we did, but when she's testing the cytotoxicity of the T cells, whether they received scrambled and antisense oligonucleotide or they received splice switching oligonucleotides, there's an improvement in the capacity of the cells to show CD107 mobilization or to induce caspase in melanoma targets if these cells are producing excessive soluble PD1. Another hit that we found was FAS. So I will be quick about FAS because I sometimes wonder if that is a true immune checkpoint receptor or a survivability gene. But what I will tell you about FAS is that very much like PD1, in early time points in non-activated T cells, you can see that FAS is about 50-50, 50% membrane-bound, 50% actually produced as a soluble decoy. And then across activation, most of the FAS is membrane-bound. So you can guess what we did. We used splice switching antisense oligonucleotides. Most of the FAS that our T cells could produce was soluble, and this gave our T cells very strong persistence benefit, activation benefit, and I will skip my slides because the principle is a bit complicated, and it took me some excessive time that I didn't plan, but you can see how when you manage to neutralize the transmembrane FAS and all your T cells are capable of doing is actually to produce soluble FAS, then they do everything better: proliferation, interferon gamma secretion. We can capture actually the extra membrane part of FAS, and it is like a לולאה מתהדקת, the extent of the process is just getting more and more the more the T cell is activated. So as you can imagine, this is something we really want to take to patients, and I'll skip some of the screenings that we did just in order to reach this important slide, and acknowledge the huge work done by my MD PhD program students Shai Tzaban, Ori Stern, and Elad Zisman. I want to mention my researchers in the lab, Dr. Galit Eisenberg, Dr. Shira Klein, and it's a wonderful occasion to thank Keren Yitzhak for long time collaboration. Thank you.
Thank you. I have personal interest in FAS as a fascinating story, so we'll continue afterwards to hear more about it. But any questions? We have time for one question.
What I'm showing is non-biased heat maps for the ones that you can see their splicing. But I must say, relating to splicing factors, which may be the same as, related to transcription factors, which may also be the problem with splicing factors, is that we use them a little bit as markers or definers of T cell state, but will be very difficult to target them for drugs.
Is a prerogative of the chair. Can I ask one quick question? We see FAS mutations and maybe it's splicing and in brain metastases from melanoma, breast cancer, and lung. And I'm wondering if it's something that you've seen or makes sense with your work?
So we haven't looked yet at tumors, but of course this is the next step we want to do, is to see how splicing affect these ligands, receptors. They actually sometimes interchange, but what I must say is that on T cells there is the FAS, but also the FAS ligand. So cis interactions are very dominant, and the tumor may have something to say about that.
To be continued. Thank you. So, moving from Jerusalem to Rehovot, our next speaker is Professor Ido Amit from the Weizmann Institute of Science. Ido?
Yeah, I'm sorry, I didn't recognize that I live in Rehovot, so, she was calling. Okay, so what I want to do in the brief time we have today is kind of discuss with you how single-cell genomic technologies are allowing us to have a better and maybe a more complete view of how the tumor and the immune system are interacting, and perhaps bring new ideas to develop better immunotherapies. So the field of single-cell genomics is young. Over the years, we've developed a lot of technology which allow us to look into the... Oh, תודה רבה. All right. Develop a lot of different utilities. First, to look at the units of biology, the cells, but then add many other features, look at the chromatin in the cells in combination with RNA, so starting the area of multiomics, look at spatial locations, so where the cells are located and their neighboring cells, enabling to look at the genetics of individual cells and how that influence their activity in vivo in the tumor site, signaling metabolism, and recently looking at how cells interact. And this is a story from Mirav Cohen, who is here now in Tel Aviv, doing amazing work here locally. And there are many other technologies on the way. Maybe I'll have some other time to talk about them, not on this short time. So what we are trying to do is really build a comprehensive data, a population scale, if you will, looking at how the immune system acts in various patients under various treatments, and combine all of these multiple layers that I described earlier into one model that perhaps can start to elucidate new and more effective direction. Again, this encompasses a lot of analytics, machine learning to incorporate all this and come with reasonable ideas. And I'll show you some of it, how it brings to new and what we think are exciting targets. So there was a very nice talk about how understanding or having better biomarkers, for example, of PD-1, which is a really amazing molecule that initiated this deep and really showcasing the potential of immunotherapy to cure cancer. So one thing, maybe we don't detect the right patient because we don't have biomarkers. The other thing is maybe we don't really understand what these checkpoints are actually doing. That's what I'm going to talk to you. But I think we all are in a clear understanding that while effective checkpoint blockade, like anti-PD-1, is doing massive effects for patients, saving really millions of lives, but also most of the patients are not responding. Even if we go to melanoma, there's huge amounts of patients not responding. If we go to other tumors, lung, breast, so on, then the responses we see are unsatisfactory, unacceptable, if you will. And what I'm going to share with you is a story driven by Ido Yofer, extremely talented postdoc from the group, with Adam and Tomer, two PhD students, where we asked a question in a slightly different way. And that is, do we really understand what our checkpoint molecules are doing in vivo? Because we have the hypothesis, have all of the in vitro work to develop the molecule, but perhaps when and how they act in vivo may be very different, and that is this story. Specifically about CTLA-4, a few words, some immunology I think is important here. So CTLA-4 is considered a checkpoint molecule. It's actually the first immunotherapy developed, and the way it is supposed to work, or the way the posters show it functions, is through this interaction over here with the CD80, CD86 co-stimulatory molecule, which in proper immune activation interacts with CD28 and activates T-cell responses. However, the idea is that very similar to PD-1, CTLA-4 is a checkpoint which takes this co-stimulatory interaction, and by that inhibits T-cell responses in the tumor. That's the idea. We had a few problems with this idea because the data doesn't really add up. and I'll show you why. If you look at anti-PD-1, clearly effective checkpoint is a checkpoint. If you have an antibody which just blocks the interaction of PD-1 and PD-L1, okay, in the tumor, what you see is a improved response, okay? But if you now change the tail of the antibodies, right? Antibody have the binding domain and the FC portion, Fab and the FC portion, and the FC portion can activate the immune response through different mechanisms, ADCC and so on and so forth. So, if you make an antibody now, a version of the same anti-PD-1, same Fab, but now you change it to an active FC, so now it will kill basically the cell that it binds to, you're basically losing the response. Okay, this is what you expect from a molecule that you need to block, okay, to have a classical blocker. But if you deplete the cell that has these molecules, you will lose the response. Okay, that's the expected results. But if now we shift to CTLA-4, which should anti-CTLA-4, which should act exactly like anti-PD-1, what we see is a completely different story. So similar experiment, same Fab, different FC. And what you can see now with the blocking antibody M1, we have a little bit of response, but very minor. But now if we add a effector tail, okay, M2A, now this antibody becomes extremely effective. So what's going on? What's the story here? How does it work? And for this, we do what we always do. We want to understand how things work in vivo. So we go to animal models and we build animal models that recapitulate the immune environment for a patient. Our single cell really allows us to align, and we know we have the same type of immune perturbation in the animal models as well as in the patients. So, oh, Michal, I don't know where she is. Never mind. So patients and the animal models are not the same. They're extremely different. Animal models are not human patient, but they can be great avatars to understand the function of immunotherapy molecules if one sees the same perturbation, the same type of trajectory or inactivation that the immune cells are going in the human tumors, see the same type of trajectories and in the mouse models. And in that case, they are good to simulate the potential function of a molecule. And so we take animal models that recapitulate the same type of immune perturbation we see in the patients, and then we build these large atlases. And here we did it for the control animals, the M1 molecule just blocking CTLA-4, and the M2A, which has the effector function. And what we see is that, again, there is very little perturbation with the M1. One would expect it, again, because it has no efficacy, whereas the M2A has a very large perturbation. 8 days after the treatment, what we see is this huge change in neutrophil infiltration, okay, and destruction of the tumor. And then if one depletes the neutrophils, okay, using antibodies, there is no effect. So neutrophils are massively perturbed, okay, with the treatment, effective treatment, but they have no effect, okay? The reason is because the immune system is extremely dynamic. And if you look, this is 8 days after treatment, you're missing the direct effect of the molecule, okay? Neutrophils obviously don't have CTLA-4 expression, no protein or RNA. They're irrelevant, that's a secondary, tertiary event. And the whole reason I told you this story is because that's what we usually do. We look at one certain time point, and if you think about patients and trying to assess the effects of molecules on patients, the ability to do biopsies or sampling from tumors is extremely difficult. To think about looking at a dense time course is practically unethical and impossible. And therefore, we have this big limitation of really understanding what is the mode of action, how does our drug work? And if we want to think which patients are going to benefit from it, how do we improve the drug, we must know how it works. So, again, what we do is we don't look at one time point, but we actually look at such immune atlases on a very dense time course. And if one looks 24 after the treatment with the anti-CTLA-4 molecule, you see a completely different story. And the story is like this. We see two main effects. Again, these are effects which are happening 24 hours after the treatment, likely direct. The first effect is this change in the ratios of Tregs to CD8. So Tregs are almost completely depleted, or 75% of them are depleted. And then this very massive change of the myeloid compartment, shifting from suppressive macrophages to more inflammatory monocytes. Okay? And just to tell you that this effect is not specific to CTLA-4. If we take other molecules which have the same type of pattern, and the pattern of CTLA-4 expression is it's extremely high on Tregs, 8-fold lower on other T cells, and then basically one cannot find it on other cells in the system. So there's other checkpoint molecules which show the same pattern. TIGIT is, again, same type of molecule, same type of behavior, very high on Tregs, lower on CD8. And when pharma tries to develop an antibody, you can see how if it develops the antibody with an FC which has effector function, it will have a drug that has efficacy, again in some of the cases, but will have efficacy. And if they don't, there's nothing happening. And all of this, again, is something that has to be really understood before you go to this billion-dollar clinical trial that you're assessing. And again, TIGIT, another potential checkpoint in the arms race against cancer, really is showing the same. And from the few clinical trials that are already advanced enough and were published, we see the same thing. Effector function TIGIT are showing good effects for patients, and the one without are very limited and not showing any effect at all. And again, that has to do with this behavior where Tregs have the highest expression. They're the one who are depleted. And when you do this, this is in the case of the TIGIT molecule, what you have are you're depleting the Tregs that have the highest level, or TIGIT of CTLA-4. They're the most inhibitory Tregs in the tumor. And then new ones are arising, but they would be less inhibitory, and when they become more inhibitory, they get depleted. So that's one effect, but that's not all the effect. And actually, the other major 2 minutes, I'm not even in the halfway of the story. Never mind, I'll finish with this. And then, all right, so this is only half of the story. Half of the story of half of the presentation I prepared, because even if we deplete Tregs by other means, so we use diphtheria toxin to deplete Tregs, we don't get the same effect as we get with the antibody. And I show you the other effect that we see is this reshaping of the myeloid compartment. And long story short, read the paper if you want the full story. What is actually happening is that the FC portion binds to macrophages, these inhibitory macrophages in the tumor, activate the FC gamma receptor, which initiate type 1 interferon response, and that leads to infiltration of monocytes, which are more inflammatory and activate the immune response, and reduce the inhibitory effect of the macrophages. So you really need these two arms of modification of the antibody to have the full effect of the anti-CTLA-4 molecule. And I think I'll finish here. It's really half of what I prepared, but it just goes to show you that in order to really develop therapeutics that are going to be beneficial for patients, we really have to understand how they're working, what is their mode of action, how they're modifying the immune system, if they're modifying the immune system. And that in reality, what I believe is that as we learn these huge amounts of data, we will be able to not only measure the immune system very efficiently, but also engineer it to our desire, and that's where we're really going with some of the projects that I don't have time to present today. But also, if we want to have these deep effects for patients which are resistant to anti-PD-1, which is again a miracle drug for a very small number of patients, we're going to have to modify not a single arm of the immune system, not only the T cells, but the multiple arms which act to suppress immune activation in patients. And since in my mind, or from the data we see, most of the patients are not what people call cold tumor, meaning they're immune neglected and the immune system doesn't detect the tumors. But most of the patients are what I call immune neutral, meaning the immune system is there, detects the tumor, but our current generation of treatments are not able to activate them sufficiently. And so I think there's great hope for having in the next several years much better and more deep responses, similar to the really miracle drugs that we're seeing for blood tumors, which are gaining more and more from the bites, and others which are getting more and more effect. So as you see, I really prepared a whole, and I'll finish here, and it was a great pleasure just to end by thanking the amazing collaborators that we have, Ronit Dahan, Amos Tanai, Nir Yosef, and others, and the super creative postdocs and students who are doing all this. And I showed the work of Oren, Ido, and Adam. Thank you.
First, I'm really sorry to cut this fascinating talk. We'll have to have a rain check for next time for the longer story. But if I can take something from this, the beginning of the story is that there are no cold tumors. There are just badly engineered antibodies, maybe. So.
No, I mean, there are cold tumors, but they're actually a very small percent. Most of the tumors are neutral tumors. So we have bad, yes. We don't have, I don't say bad, I don't think we don't have good enough.
Can be improved.
We don't have good enough.
But I'm wondering if by this you can explain if we compare the anti-CTLA-4 and the anti-TIGIT to anti-PD-1, that was, even though everyone can agree with you that it works as a miracle only on a subpopulation, but still, the others did not do that. Can we explain the less efficacious, and again, CTLA-4 less, and TIGIT was very disappointing compared to even to CTLA-4, not to speak about PD-1. Can we explain also the toxicity of anti-CTLA-4 by changing this FC?
I think the toxicity may also come from the same mechanism. CTLA-4 is expressed on Tregs mainly in the tumor, but not only in the tumor. So I think there are some, I would say, depletion of Tregs from other sites, which are likely initiating unwanted side effects that are happening, which also reduce its efficacy, right? Because you have to limit the amount you're giving to the patient. So, definitely, the more we understand the mode of action of drugs, the more we can design them to limit the off-target effect and increase the on-target.
No, think about
verify that you have the right
You're absolutely right. So well, it, I don't think it's acting CTLA-4 as a checkpoint, right? We're seeing more and more, and you'll probably talk about your drug, that some of the more or some of the effects of molecules are not the effects we actually designed in the beginning, but some of their side effects can be even more effective than what we designed initially. And we see that only when we go into the patient and start to understand what the drugs are actually doing. So in this case, as you're saying, there might be molecules which have a more narrow expression, which it can really target and deplete a very specific inhibitory cell, and in that case will have better efficacy and less off-target effect. Because as you can imagine, depleting the CD8 with CTLA-4 is not necessarily a good thing, right? So there is good effects, and it's all mixed together because we really don't understand fully the activity of the drugs that we design and where is actually the benefit coming from which of the many arms that they have inside the tumor, which is a very different environment from how we test them in vitro. And so a more realistic model that allows us to model and measure and design the multifacet of the drugs, the better we're going to have drugs that when they go into patient, they will do more of what we designed them to do and less of, by chance activity. And we talked on the taxi on the way on several other such drugs. Yeah. Yeah. So what Saf is asking, so T cells in the tumor are basically, in some patients, actually a very large number of them become exhausted or dysfunctional. And there is a big debate in the field, is it beneficial to deplete them and perhaps make more room for T cells that would be cytotoxic and fight the tumor? So from the experiment I showed in the beginning, I don't think that's maybe if there's something really directed at the very terminally exhausted, maybe there would be some effect because there are some studies that are showing that they are by themselves slightly or inhibitory in some of their aspects. So perhaps again, if we can find a really very narrow target that will be exclusively on these cells, that there is some beneficial effect that would be to remove them. They probably are doing, I don't think it's the capture of space as much as they do secrete molecules which are not, let's say, efficiently activating or even somewhat suppressing the system. So that's likely the case, but again, there is always the fine balance of what are the other elements that you're affecting and would they contribute or inhibit the.
Thank you, Ido. So we stay at the Weizmann Institute of Science, now with Professor Yifat Merbel.
Okay, so good afternoon, everyone, and it's great to be here. And what I would like to take us through today is downstream to alternative splicing, downstream to gene expression, and start to have questions that are related to what we determine as the epiproteome. I'll, of course, explain what it is as we go along. And so when we think about the paradigms in biology, and of course this is no longer true, we understand the complexity of biology is much deeper than that. But by and large, when we talk about the therapies, the antibodies that control responses to cancer and so on, we talk about proteins. be it antibodies, be it targeted therapies, we're targeting proteins. And there is a lot of efforts that are invested in understanding the landscape, not only of mRNA, but also the proteins in the tumor microenvironment, in the tumor themselves. And heavily we rely on a technology that is called mass spectrometry. And this is allowing us to really zoom in on all of the list of content of proteins that are being expressed in different tissues. It may be different cell types and so on and so forth. What my lab does and where we focus is primarily what happens downstream to it. Meaning after a protein has been synthesized, it undergoes a lot of changes, post-translation modification, changes in activity, changes in cellular localization, and all of these together ultimately determine what the protein does. Now, it's not that everyone in the audience is aware of it. Why is it that we don't understand it? Why is it that it's not utilized yet in the clinic? And the reason is that the technologies are basically lagging behind. And so my lab primarily focuses on the PTM, which stands for post-translational modification and proteolysis, but I'll talk today about PTMs alone, and we define all of these changes that occur onto proteins as the epiproteome. And so the idea is not only to now generate novel questions that we will address at this realm, but to also tackle them in a translational manner that may be utilized to harness and help in patients. And what I'll talk about today is only one story. Hopefully the time will suffice. And then we focus primarily on what happens in this interaction. So we had a very nice introduction by Ido. I'm going to talk about CD8 cytotoxic T cells and basically how they recognize the antigens that are presented on the surface of tumor cells. And this is where we'll dive into, and we harness a new technology that we developed to zoom into the epiproteome. Now, primarily, this is not going to be sufficient. Clearly, what I'm going to show you is only a first step, but our question and what we had in mind is how can we utilize this understanding of post-translational modification to better understand this interaction and better exploit the ability to harness the immune system to target the tumor cells. Now, when we talk about post-translational modifications or PTMs, there are more than 200 different types existing in our cells and bodies. It may be phosphorylation, acetylation, ubiquination, isogenylation, UFMylation, and so on and so forth. I won't continue. And the problem is that they're very different in nature. So we have biochemical modifications, we have modifications that conjugate a protein onto other proteins, and that really limits our ability to look at them globally and analyze them globally. And so just to give you an example for the computational limitation, basically if we want to look at changes in PTMs, okay, in post-translational modification of a protein, usually when you send a sample to mass spectrometry analysis, or be it just one protein, it will be cleaved in a very particular manner. You will have trypsin and it will cleave it in an arginine and lysine residue. So we can a priori just assess what are the peptides that are going to come out of the computation. Because you'll have all the peptides within a protein that are either ending in lysine or arginine. What if we wanted to now enhance it to look not only at the lysine and arginine cut, but also on peptide that had a modification? Let's say phosphorylation. So this is what happens to the search space. Basically, if you have tryptic and phospho sites, only 3 possible amino acids to have a phosphorylation, we're staying at the same level of landscape, theoretical landscape for the mass spec for the potential peptides. But if you now have non-specific cleavages, which are what we see when we isolate antigens that are sitting on the surface of cells because they have any termini. They're not only cleaved by the cell by lysine or arginine. Now we have to identify peptides that are more diverse. And if we add on top of these non-tryptic cleavages, the modification, we enlarge the search space in orders of magnitude. And this is only for phosphorylation. What if we have now 200 other types of modification? And this is what we set out to solve, because this problem is very well known as the dark matter of the proteome. I'm going to talk about antigens. Everything I'm going to be talking about is related to any protein in the cell. It may be an antibody, it may be a transcription factor, or a cytokine. The dark matter of the proteome is the 50 to 70% of data that is running in the mass spec. There is a physical entity, a peptide, that was not detected because we didn't provide the right reference proteome, meaning we provided only the amino acid sequence without the possible mutation, natural variability, PTMs, and so on and so forth. So each and every one of you who submitted a sample to mass spec and didn't ask in particular for a specific modification to be detected could not have found it. And that means that we have treasures, tremendous amount of data that we can reanalyze and ask what is hidden in the dark matter. And so this is the tool we wanted to basically generate, which would allow us to really detect more than we previously thought, and to go beyond the small fraction that is currently detected by mass spec. In order to do so, Asaf Kassen in my lab, and this was recently published 2 months ago, developed a tool which we called PROMISE. It's a big name, and we hope it will bring a big change. PROMISE stands for Protein Modification Integrated Search Engine. And the idea is that instead of using only the reference protein, there is one in UniProt, for humans, we can actually generate an infinite amount of reference proteomes, be it by variation, post-translation modification, and so on, to see all of the diversity. Now, of course, we don't want to generate more noise. We want to keep accuracy, and we want to keep speed, and so on. So there are a lot of conditions and a lot of steps that are going into this calculation to make it a really useful tool in not just generating noise, but actually increasing the signal. I won't go into it. These are mass spec specific steps. But just to say it simply, what we can now do in a manner that is very robust is detect up to 30 different types of modification. You can look for your personal, ADP ribosylation, ubiquylation, sumoylation, phosphorylation, acetylation, methylation, and so on. And we do so in a manner that not only keeps the running and the speed really better than any existing tool on the market, but also keeps the accuracy and the depth of the read really intact. And what it also offers is that I don't need to run any new sample if I just want to go back to any sample that was analyzed in the last, let's say, 4 or 5 years, go to the raw data, reanalyze it by PROMISE, and see what was missed there, because the data exists, it just is not transformed into information. And again, we're enhancing the types of modification we can detect. It's now actually more than 30 PTMs. And so just by way of introduction, when we're thinking about a first biological question to address, this is going to be looking at the antigen landscape, the antigenic landscape, the whole realm of neoantigens, mutations that are driven to generate novel antigens. We speculated that perhaps if you have an altered cellular state due to cancer, due to KRAS activation, due to constitutive signaling or ARP gene addiction, we would actually change the state of signaling in cells. Why don't we try to look for modified antigens instead of neoantigens that are just mutation driven? And perhaps because the cancer can mutate a lot, that's its power. If we target these addictive signaling pathways, we actually have leverage to target the cancer in something that it cannot get rid of. Okay, an oncogenic pathway activated for KRAS cannot get rid of the phosphorylation that are downstream to KRAS, and that will ultimately go to the surface. And so just by really not going into too many details, we have 2 regions that we're going to sample here to make it very simplified. When an antigen is bound to an MHC molecule, we have usually around 11 amino acid long. There is a range, but to simplify things, we're going to look at the 11 amino acid long molecules, where the 2nd and 9th position of the antigen determine the anchoring to the MHC. And the middle region is related to what the T-cell receptor would recognize. Okay, and we're separating these 2 classes, and that will become important later on. So first, what we wanted to do is look at mice. Clearly, it's just a model system, as Ido mentioned. Nevertheless, you can learn a lot from it. So we analyzed the mouse data of the immunopeptidome, again, antigens that are sitting on the surface. And we did this with the cell line, which is MC38. And we identified a whole lot of antigens that were modified by these specific modifications that are indicated here across the MC38 cell line. And we then asked what is unique to the MC38 tumor cell line that does not exist in any of the other tissues that we had immunopeptidome for. And we identified a class that was unique to that cell line. So presumably we can identify cancer-associated antigens that are based on modification in the mouse. And so we then continued to follow up and validate this. I won't go through all of it. It's published in the paper. And we could show that we indeed have bona fide binding to MHC class 1. And not only that, but that in some cases, the modification itself can actually change the binding. So if we look at this example, the red color here is the modified state. The gray is the unmodified peptide, the exact same peptide. You have here a dose curve for the binding to the MHC, and you can see that the modified state is actually binding better. In some cases, the modification will not have an effect, and in others, it can actually attenuate or inhibit binding. And so then the question is what about the human immunopeptidome? Can we identify something there, and can we come with or highlight principles that are underlying this modified state of modification? I'll explain this graph. Okay, so zoom with me in on the 11 amino acids that you have here. So in this circle, every line, as you go like this, is a peptide, okay? Every gray line. And it's arranged merely by the amino acids here in the circle, and in the rim, the type of modification. The reason it's so heavy is because you see here more than 11,000 peptides. I could have provided a list of peptides in a table, but we would be able to look at only a very small portion. What it also allows us to look for is then motifs, meaning what comes up more than we would expect by chance. And I highlighted here one example of that sort. You see this box here? This tells us that many peptides had on position 4 of the 11 amino acid sequence that was eluted from the MHC, it had a serine in the position 4 that was phosphorylated. Okay? Now, if I ask you what else is interesting here, you wouldn't be able to pick it by eyes. But what we can do is just analyze it computationally and look for these motifs. So on the left here is the example I showed you. If we take all of the peptides and we ask how many of them were modified on a serine residue in position number 4, you see this red distribution. That's our distribution of the modified state. And we can compare it to two different backgrounds. The IEDB, that's the epitope database with all of the epitopes that are known to be deposited there, not just by us, but worldwide. And also the blue background, which is our specific database that incorporated a lot of different samples and so on. And what you can see clearly, if you subtract this, you have a distribution that is above and beyond what would be expected and highly significant. We're not talking here a small number of peptides. These are very large amount of peptides. So we have an influence of serine phosphorylation in position number 4. We went back to the literature. We asked, is it known? So 6 different papers, bottom up, focused on one peptide that they characterized. And when they realized that if it's phosphorylated on serine number 4, it actually drives immune evasion, meaning the T cell recognition was lost because of that negative charge on the phosphorylation. So we said, okay, we're not inventing here the wheel. There are evidence from the wheel that exists. If we look at the technical modification, what is a technical modification? Oxidation on methionine is something that happens in the mass spec as the peptide flies. So it's a modification that is recognized by the mass spec, but as you can see, it doesn't deviate from the background alone. So what it means is that this modification is not going to inform about either MHC binding or TCR recognition. We can transform, let me explain this because I see it's missing. If we subtract the red from the blue, okay, we get a heat map. Imagine that I have this heat map and I'm subtracting this. This will be blue because it's lower, and this will be red because it's higher, okay? Now we transform this all across modification, okay, and amino acid types. So if we look at the serine phosphorylation, which is here, which is our example, position number 4 is red, and the position number 1 is lower. What also comes up of this global analysis is that, for example, anyone in the world right now trying to generate an anti-cancer antigen, anti-cancer vaccine, and will design an antigen will never choose a lysine residue in position number 2. I can tell you, in the prediction tools, lysine is never selected. Why? Because it seems to be not preferential for MHC anchoring. But what we actually see in this data, there is plenty of lysine. Lysine is K. There is plenty of lysine residue in the anchoring position. They're actually enriched, but in a modified state. And that tells us that there are specific types of antigens that would actually be recognized only in the modified state of the lysine residue. We can also take this and now do it for a haplotype specific, because when we treat with either antigen, cancer vaccine, TIL therapy, and so on, each person has their own haplotype for the HLA, and we do see motifs that are deviating, that are specific to each haplotype. I won't go into it, but this is very important and very interesting in the context of autoimmunity, which is not driven by mutation, but rather by changes in antigenicity. And so beyond the ability to highlight novel motifs, we can then go back and revisit a lot of the cancer-associated genes and antigens that have been detected in the past and examined in the past, and ask, can we identify for any of the tumor-associated genes antigens that have been not detected due to a modification state? So here I give an example for BRCA, which has a novel antigens merely based on the modification. You can have these from really interesting pathways, RASA, BRCA, and so on and so forth, suggesting novel antigens to be detected and also targeted by therapy, because for some of these genes, we know that they're going to be tumor-specific. And to just take us back to a really fundamental biologic question, if we see these modifications on the surface, just to remind ourselves, anything presented on MHC class 1 actually went through the proteasome. So we asked ourselves, we were intrigued, can we identify this modified peptide in standard proteomics in the cells? Because if we detect it on the surface, it should have been modified, went through the proteasome, and carried to the surface. And let's focus, just because of the limitation of time, on this peptide here from this molecule, from this protein, and you can see these specific sites on this peptide, and we asked ourselves, can we detect it in a breast cancer tumor or adjacent controls? And what you see here is really phenomenal. I'll explain why. The blue dots are adjacent tissue. Each dot is a peptide that was detected exactly as such. This is not just site-specific, not just peptide, but it's exactly this one that was recognized on the surface. And each dot here in the orange state is actually in the cancer, meaning we have a state of modification that was generated due to the altered cancer metabolism, carried to the surface, and highly significant, and may presumably a target for cancer. Now, this really goes to show that if we track changes inside the tumor cells, and we also associate them with what we see on the surface, we have a leverage to really identify valuable targets. And so we really believe that modified antigens should be examined not only in the context of cancer, but also in other human disease. To summarize what I've told you so far is that we managed to basically not only enrich the HLA immunopeptidome, but we also devised the first atlas of the modified immunopeptidome. And clearly I cannot go over a lot of modifications, but there is really golden nuggets in there that really tell us something new about immunology. Furthermore, we could show that the PTM state can really alter either the MHC binding, like the example of the lysine residue, but also TCR recognition. We have other examples like the phosphorylation on serine 4 that are not known to the world today. We can also show that for a subclass of this, and we've done this experiment in mice, we can actually elicit tumor reactivity against them, meaning T-cell reactivity. I should say explicitly that having a molecule bound to the MHC doesn't tell us anything about the immunogenicity of that antigen. So that's a holy grail that we are now trying to understand, whether there are also rules for which type of modifications cause immune evasion versus which type of modification may actually activate the immune system. And we uncovered thousands of putative modified antigens and PTMs that really offer novel ways to therapy. But if we now think about this as a novel class of neoantigens, that we can extrapolate it to any neoantigen we can think about, be it in the context of autoimmunity, be it in the context of viral infection, oncogene, and so on. So essentially what we're providing here is a novel paradigm by which not only the amino acid sequence should be examined, but also the amino acid sequence with the combination of the modified state, which will in fact be altered due to biological states in vivo. And with that, I'll just go to highlight some of the questions that are really left unanswered and really open for us, a whole world. I've mentioned the question of identifying the rules for enhancing immunogenicity. There's also a lot of crosstalk between the tumor and immune that is mediated by PTMs, not necessarily just on antigens, but other molecules, and this is something we're exploring right now. Crosstalk between different PTMs. We revealed some rules that we see that go along together. This field is completely untouched yet because we didn't have this analysis across 30 different types of modification at once. Of course, spatial and temporal resolution, understanding how PTMs change that. And I think for the future, what we want to think about is just like we have mutations that are driving disease, we may have pathogenic PTMs that are driving human diseases, and with such tools and many others, we will be able to explore them and look into them. With that, I would like to really, again, mention Asaf Kassen, who was the main driver of this work, his collaborator Aaron, and Merav and Matthias, who were responsible for the biochemistry and the in vivo, Asaf and Aaron for all the computational part, and of course our collaborators and the finding agencies. Thank you very much.
Thank you, Merav.
Ifat.
I'm really enjoying this day. I'm learning so much from every talk. I totally second Michal Lotemir that we're more to listen than to speak. So questions, yes, Moshe. Louder.
I think so. In a different study that we have on proteasomal cleavage, which is an upstream event to antigen presentation, and is going to come out soon in Nature Cancer, we actually show that is the case. So one of the ways that are being adopted and hijacked by tumor cells is to actually change the HLA landscape.
Can you repeat what he said?
So all great questions, and the answer to the first two are no. I can hand wave about what I think is going to happen, and I think yes. I think if you harness this, and this is what I try to allude to with regards to oncogenic addiction, we can actually harness this to target specific targets. Not only that, but we talked before, you can even think about, giving a drug, HDAC inhibitor, phosphatase, and so on, and come with a hammer because to be able to predict which modifications and what is going to go to the surface. Regarding the immune tumor interactions, absolutely, but we haven't done it yet, or other immune cell types, and MHC class 2 is definitely something that you can now go and do a parallel experiment and study, but we haven't done it yet. Thank you. So we haven't done so, and one of the reviewers asked for he wanted to have PROMIS glyco, and we said, okay, we'll take it on as a challenge. Glycosylation is a whole world by itself. Mark Cobbold has identified both antigens that are glycosylated, but also the MHC molecule, and that in fact affects both the MHC binding and tumor and TCR recognition. So the data is there. No systematic study yet because of a limitation of the tool. Hopefully we will get there.
Thank you, Ifat. Amazing talk. Next speaker from the Hebrew University of Jerusalem, Professor Yinon Ben-Arian.
Okay, thank you very much for the invitation. I studied in this school, so in the medical school of the Tel Aviv University, so it's always good to come back. We are, what we are doing in the mountains, in Jerusalem, is daily life. We are studying signaling pathways in animal models. And recently we got attracted to a specific disease for which there are very good models, and this is AML, acute myeloid leukemia. Now, acute myeloid leukemia is really an intriguing one because there is a myriad of genetic and pathogenetic mechanisms to drive different sorts of the disease. But it also is challenging therapeutically what we call unmet therapy needs, because most of the patients nowadays, even with modern therapy, relapse after treatment. And here you see, can you see that? Yeah. You see the relapse rate, basically the 5-year survival of all patients, including young ones and old ones, is only about 27%. So there is still a lot to do in the disease. So just about 4 or 5 years ago, the FDA approved the first targeted therapies for AML. Before, all the patients were treated with chemotherapy, which was developed about 60 years ago. Still, it's the main treatment for AML. But these drugs were very encouraging in a way because now you can actually target AML mutation, and there are two types of major mutation in AML can be targeted. One of them is a surface receptor called FLT3, and the other is an enzyme, isocitrate dehydrogenase, IDH1 and IDH2. Together they comprise about 50% of all the AML mutations, and many drugs have been targeted to these ones. Here is the first one that were approved. But what we can see, although it had a great promise, that the success rate is kind of limited because they mainly, if you look, they provide a few more months of survival to the patients. So that's a common occurrence in cancer therapy, that most of the target therapy actually do not last long enough. So we need to think of something else, and instead of targeting the mutation, the cancer driver themselves, we thought, and many others are thinking likewise, let's target what we call cancer vulnerabilities, weak points within cancer cells. And indeed, the cancer home is pretty fragile, and that's because of many mutations, energy requirements, metabolic, and so on, so it's kind of fragile. And it's very much dependent then on survival protection pathways. So what you need to do is to disrupt those pathways, to interfere with them. And the most successful drug actually in AML therapy as of today is a drug that does exactly the same thing. It actually compromises survival of leukemia cells, and it's used for many other hematological malignancies as well. So usually it's not working by itself, like many other cancer drugs. You need to complement with something, but if you complement it with hypomethylating agents, for example, then all of a sudden you see a very nice response. The pointer here is very sensitive. So, but also this drug this therapy doesn't last very long, because after a while, so what, actually, let me tell you, what the drug, which is called venetoclax, is doing is interfering with the binding of BEAM to BCL2. And by doing that, it cuts the apoptotic cascade, which is initiated by BEAM. Now, after a little while you inhibit BCL2, another protein is upregulated in the same leukemia cell, and this is called MCL1. MCL1 can also bind BEAM, but it cannot bind venetoclax, so it's actually immune against venetoclax. So this is nice, but let's think of other ways to actually compromise survival, and the other way is using the p53 tumor suppressor. Now, AML is one of the few examples in which p53 is non-mutated, wild type in nature. However, it's usually inactive, and it's inactive because of a lot of antagonistic molecule, antagonistic protein that work around it. And the most important one is MDM2. MDM2 is a ubiquitin ligase for p53, also blocks its transcriptional activity, and it's also target gene of p53. So when you try to activate p53, MDM2 comes on and actually blocks the activation. So a lot of companies nevertheless try to develop drugs that interfere the interaction between MDM2 and P53. Roche was the first one, so this activity is going on for 20 years. But so far, the molecules which, the small molecule which have been generated are only working kind of modestly, and they are very cytotoxic, so a lot of them cause thrombocytopenia, so they are not in use yet. Our approach was different. So our approach was to try and activate P53 through a signaling system which we actually identified in the Wnt pathway. And this is an enzyme called CK1 alpha. It's a protein kinase, which normally we found that to phosphorylate beta-catenin. But when you inhibit CK1 alpha, either genetically or now pharmacologically, you induce P53 activation. You can see that here, homozygous CK1 alpha knockout in the mouse gut. You all of a sudden see the nuclei are full of P53 expression. So based on this observation, we started to develop CK1 inhibitors, small molecules, and succeeded to produce a large series of inhibitors, the first one of its kind because there are no other CK1 alpha inhibitors known. And this is a co-crystal structure of one of the inhibitors with CK1 alpha. You see it's catalytic, sits in the ATP binding pocket, and so on. So what does it do? It's actually, the inhibitors mimic very well the ablation of CK1 alpha. They stabilize beta-catenin, you see that nicely stabilized, but they also stabilize P53. And here you see, even in low nanomolar concentration. But actually, the best inhibitors that we found, the best inhibitors to activate P53 had extra activity. And what they do is they co-target two transcriptional kinases called CDK7 and CDK9. Oh, sorry. These are necessary to phosphorylate the C-terminal domain of RNA polymerase II, which is important for transcriptional initiation and elongation. There is an antibody that you can look at this phosphorylation event, and you can see that this is oral treatment of leukemia mice and harvesting the leukemia cell after 5 hours, whereas you started in the vehicle or the control with a lot of phosphorylation signal. After a few hours, you don't have a trace of it. Why is it important? Because this transcriptional block interferes with the transcription of many important leukemia drivers. So I mentioned something about the MCL1. So you see, that was MCL1 before and after treatment with one of the molecules, you don't find a trace of it. And what is also interesting here, MDM2, I told you, is a negative loop, negative feedback loop of P53 activation. Now you don't have any transcription and translation of this protein, so P53 can activate it to a larger extent. Now, these transcriptional events are not limited to MCL1 and MDM2, and indeed, if you look at a whole bunch of oncogenes, and most of them are transcribed to a machinery called super enhancer, you can see that the treatment actually abolished most of them. This, on the left, you can see super enhancer. Super enhancer can be identified by an algorithm of Rick Young, and beyond the inflection point, anything here is super enhancer. So when we transform normal bone marrow cells with MLL-AF9, about 120 super enhancer are newly born in the transformed cells, and most of them can be abolished by this A51 treatment. You see, there's very little left here from all these super enhancer, and the one that we think about, MEEK and MEEB and RANKS2, all kinds of non-cancer drivers. We can also visualize the activity of our small molecule with regard to super enhancer, because super enhancer are thought to operate within a hub, a condensate called liquid-liquid phase separation condensate. And if you look carefully into these slides, so sensitive that I'm afraid to touch here. Oh, sorry. You can you can see that within the nuclei there are green. Okay, yeah, that's much better. There are green dots which represents those liquid phase condensates, and once you treat the leukemia cells with the inhibitors, actually most of the dots are done. So why is it important? Why do we bother to abolish super enhancer? Because cancer cells have what Phil Sharp initially called transcriptional addiction. So they are very sensitive to perturbation in the transcriptional machinery. Transcription of major oncogenes works within those hubs as super enhancer, and once you abolish them, the cancer cell, or leukemia cells in our case, are the first to die. Okay, so we tried some of these molecules in animal models of leukemia. This is a very aggressive leukemia, so you see when, if you don't treat, they die within a month. If you treat, all the mice respond, and some of them are even responding for long term. In fact, they are cured. We know that because if we harvest cells from the leukemia after treatment at this stage, about 90 days, and transplanted into lethally irradiated mice that are actually dependent on the transplanted cells, we see that all the mice actually are alive for long term, meaning that first of all, we didn't compromise the normal hematopoietic precursor. Second, we don't see any evidence of the disease. So either we killed all the leukemia progenitors, or there is something in the bone marrow of the transplanted cells that prohibits the relapse of the disease. Actually, recently we found that we don't know about the first mechanism, whether all the progenitors are killed or not, but we find evidence for the second option, meaning there is something in the bone marrow that we're transplanting from those rescued animals that prevents the relapse of the disease. And what is this? You can imagine these are T cells. So we take mice that have been cured or so, long-term survival, and we re-challenge them with the same leukemia. And you see, most of the re-challenged mice, without any treatment, based only on the presence of those T cells, survive long-term. Now, if we deplete from these treated animals the T cells, now you see that most of them are actually dying. That's the green line. Meaning that, in fact, we generated memory against the specific leukemia, and this is a specific memory because if we challenge those rescued mice with another type, another leukemia type, they all die for leukemia. So they have memory to the specific leukemia, which is helping to prevent relapse of the disease. So this is just some summaries of what I told you. The unique properties of these small molecules, they have two kinds of activity. First of all, they block CK1 alpha, but this is sufficient for blocking, for activating p53 and treating certain pre-malignancy like myelodysplastic syndrome, but not AML, because in AML, we have, first of all, MDM2, which is very active and prohibits the activation of p53. And we have a lot of protectors that come by, like MYC, MCL1, and which protects the leukemia cells from apoptosis. If we come with a molecule now that has two arms, both the CK1 alpha and the transcriptional kinase blocking, then there's no MDM2, you can activate p53 to the maximum, and you don't have cell protectors, so basically the cell have to die. Now, just to remind you that with all these activities, the transcriptional activity and CK1 alpha, we need something else, and the something else is combat T cells that are engaging with the leukemia cells. They preserve memory, so now when they are sitting still in the bone marrow of the treated mouse, with the memory, they prevent the relapse of the disease. One of our molecules went into clinical trial recently, and the results are pretty encouraging. What you see here is results of individual patients treated. And let me say, the conclusions were that the molecules the ones that we tested is pretty safe. The other ones, we have even a biomarker, which is called MYC1, is actually p53 target, which we can measure in the serum of the patient. And the most exciting thing about it is that a group of patients greatly benefit from this treatment. This group of patients are leukemia patients that have been treated massively with a lot of different drugs, but all of them finally got this venetoclax. Yeah. Venetoclax in combination with hypomethylating agents. So they are and failed to respond to venetoclax. Now we're coming with our molecule, and all out of the ones that have been treated so on, some of them respond preferentially. All the responders have one unique property. They carry a major AML mutation, which is called RUNX1. RUNX1 is actually a transcription factor, and the mutation is a loss of function mutation. We don't fully understand how it operates there. But after venetoclax treatment, about 40% of the refractory relapsing AML patients carry this mutation. So it's create a cohort which is nice and big and ready for a phase 2 trial. So the study was done by a few people, 3 students, and Irit Alkalai, who is my lab manager, and Walid started it. He actually was the first one to test and to even establish the right models. Avantika is still, Walid is a postdoc now. Avantika and Eric are still working on the lab. Eric is the one who is responsible for all these immune phenomena that we see now. And Avantika is the one that identified the epigenetic alterations which are induced by this model and by the treatment. And we have great collaborators, Eli Pikarsky, with whom we're working for many years, and Moshe Oren from the Weizmann Institute. And thank you for this.
Thank you, Inon. Yes.
Yeah. Right, so we looked, this is interesting, and the patient we had are not the ones that have translocation of RUNX1, but point mutation. So the translocation are a bit more complicated activity. The point mutation is simply loss of function mutation, and so it tells you. So we don't know whether translocation have any significance in this, but maybe we should look into ALL nevertheless.
Along the same lines, do you know anything in solid tumors? Is there any relevance?
So the set of molecules have many interesting targets. Some of them, like MYC, is prevalent. You can find in many different tumors. And indeed, if you there are some models that we started to analyze with. One of them is colorectal cancer, which is very much dependent on MYC. Interestingly, in colorectal cancer, those inhibitors don't work by themselves at all. But if you combine them with MAP kinase inhibitors, all of a sudden you see a dramatic effect. And this is interesting. Synergistic effect. Yeah, none of them works by itself. And this is interesting because one of the adaptation mechanisms to the therapy with these molecules is MAP kinase activation. You see that very readily after treatment. So you have to curtail it to actually suppress the MAP kinase pathway in order to allow the molecule to work in certain types of cancer.
Else? No, thank you.
Yeah.
Last but not least, here from Tel Aviv University, Professor Vered Adler-Karavani.
Thank you very much for the opportunity to present here. I'm really excited to tell you a little bit about glycans, sugars, and how our diet could affect and perhaps explain some of the risk factors related to cancer. So we all hear about our diet. It's good to eat that, and it's not good to eat that. And in general, we know that vegetarianism is related to more healthy lifestyle, while red meat consumption, and particularly processed meat, is related to cancer risk. But why is that? And there are some, of course, recommendation to limit red meat consumption and processed meat by many agencies that work on cancer. And there are several explanations that have been described, including high energy, fat diet, heme iron, TMAO, etc. But none of these explanations are really specific for red meat. For example, some of them also appear in fish or in chicken. So none of them really explain why particularly red meat is associated with cancer. And we know of a new disease mechanism that is involved in a sugar and antibodies against it, and this sugar is Mu5GC and anti-Mu5GC antibodies. And I'd like to convince you that this perhaps is the molecular link to cancer risk and red meat. Let's go back just to think about sugars. We all know that all the cells are covered with a dense layer of sugar coat. And if we zoom in, we see that this is, in fact, long sugar chains that are assembled inside the cell and delivered to the cell surface. And the glycosylation machinery is not a template-driven process, but rather a complex metabolic pathway of enzymes that are taking one sugar unit at a time to build these long sugar chains on the cell surface. And in cancer, these modifications change, and we see a different sugar coating on cancer cells. But we also know that, in particular, at the terminal edges of these long sugar chains, we see the cherry top sugar, which is called sialic acids. While in mammals, we see two types of these sialic acids, chocolate vanilla, I call it, or you can see it in purple and blue. Humans lack the ability to synthesize this blue sugar called Mu5GC. This is because all humans lack the enzyme CMAH, or CMP-Mu5AC hydroxylase, that adds a hydroxyl group to this type of sialic acid. So all mammals express both types of these sialic acids, while humans express only the Mu5AC, the human sugars. As a result, when we consume red meat, we also take up this non-human sugar, and it changes the sugar coating of our cells. And although the sugar coating of cancer is different, it becomes even more different when we consume red meat, changing the terminal sugar into Mu5GC. And that results, in fact, in the alteration of all the sugar-coating glycans that we have on the cell surface, and this sugar becomes immunogenic. Although the only difference is a single oxygen atom, all humans that we have investigated thus far have a complex immune response against this sugar, against, in fact, multiple presentations of these sugars. So we have a collection of antibodies against Mu5GC that is accumulating on our cells, particularly on cells that divide rapidly like cancer cells. In mice, we have a mouse model that, like humans, do not express the CMAH gene. And we have shown in many studies that there is a complex disease mechanism termed as xenosialitis that combines the presentation of the non-human sugar in the context of self on the cells, and by that also generates an antibody response that meets this non-human sugar in the context of self. So there is an ongoing war and generally inflammation that had been shown in mice to increase the risk of carcinomas as well as atherosclerosis and various other diseases. And in humans, we also see Mu5GC staining, particularly in many types of cancers, as you can see here, the brown staining compared to the control. And this, of course, resembles the new 5GC presentation on multiple glycans and the multiple antibodies that recognize it. In a limited study, using the Nurses' Health Study, tracking women over 30 years, we tried to see whether the total response of these antibodies could be associated with cancer risk. And indeed, we see that those individuals that have high levels of antibodies against this dietary sugar are associated with cancer risk. But we couldn't find association to the diet. So with food frequency questionnaires, when people get a questionnaire of saying, how much red meat do you eat? This much, this much, this much, or this much. And you know, people tend to lie. And when you have, when you have one questionnaire in people that have been tracked over 30 years, and you have just one question per year, it's not surprising that the association could not be found. But really, what we know is that we see an association between these anti-New5GC antibodies and cancer, at least in limited samples in human, but also in mice. And we also know of the many studies that combine the red meat diet and cancer. But really, can we match the red meat consumption to anti-New5GC antibodies? Does it really mean that if you eat a lot of red meat, you will have a lot of antibodies? And that could maybe close the circle to combine this cancer risk. To address that, we first looked at the world population, and you see my friend here from South America, Dino, that asked me this question, and just for her, I did this experiment. We looked at red meat consumption across the world, and then we asked what would be the colorectal cancer risk. And sure enough, in countries that consume high levels of red meat, you see that there is a direct correlation for colorectal cancer risk. And if you take the two extremes of the quartiles of countries that consume low levels versus high levels, you see that the separation is just robust. And in countries like Sri Lanka, India, and Africa that have low consumption of red meat, you also see lower cancer risk of colorectal cancer. And in countries like Australia, USA, Europe, and South America, you see higher cancer risk. In particular, we also noticed some gender differences, but only in the countries that consume high levels of red meat. We didn't know what it meant at that point, but then we looked further into this to investigate that. One of the countries that consume high levels of red meat is France. And in France, we had a very unique collaboration, which is called Nutrinet Santé, that had an online questionnaires of individuals that had willingly reported over 24 hours everything that they consumed. And they did that on multiple days, up to 30 days. And that included weekdays and weekends. And also in that particular study, they also confirmed in the population that of the individuals that had reported that those French people that consumed high levels of red meat also were associated with higher colorectal cancer risk and breast cancer as well. So we asked in that particular cohort, when we have a very detailed information of the diet, could we quantify New5GC in the diet, and could that be associated to the level of antibodies that these individuals have? So first thing first, we received all the French delicacies into my lab. We didn't taste that, but we did quantify New5GC in those food items, and we see the levels of New5GC in red meat and in dairy. And then we went back into the population that we had at the time, close to 20,000 individuals that had reported at least 6 days. We divided these 20,000 individuals into 4 quartiles, those that consume high or low. And then we divided, then we confined it to those individuals that had at least 18 days of reported 24 hours. And of that, we took the age group of 45 to 60 or over 60, and we took 10 individuals representing individuals from those groups for further investigation, of course, men and women as well, in order to look at the gender bias. This is a distribution of New5GC in the 20,000 individuals. or 16,000 that we had this over 18 days. And you see the distribution of new 5GC consumption in the 4 quartiles, those that consume low versus high, and in the age group of 60, which we had just a quartile 1 and 4. Now, we, of course, wanted to measure anti-new 5GC antibodies. And that is a key question because you have, as I explained, many different presentations of new 5GC on the cells. So which type of antibody should we measure? Should we measure just one type? Would that even tell us something? So to do that, actually, we designed a method to first look at the average overall response using the CMAH knockout, and I will not go into the details, but we can measure an average response against multiple presentations of new 5GC. And using this technique, first we looked at the gender differences in the age group 45 to 60. We see that men consume higher levels of new 5GC. We also know that in general, men consume more food in general. Their BMI is higher than women. And that was mostly attributed to the red meat consumption, but not different in dairy of cow or goat and sheep. And then when we looked at the average antibody responses, we see that men indeed have higher anti-new 5GC response compared to women, as we saw in the World Cancer data. And in the age group of over 60, we saw a similar trend, although it was not statistically significant, the numbers were higher in men compared to women. And this was a smaller group. Then we also have the technology of glycan microarray, where we can print different presentations of the glycans with either terminal human sugar or terminal non-human sugar, and then we can measure the specific reactivities against specific new 5GC and new 5AC glycans. And using this technology, now we take an average response and we the actual responses against multiple presentation. We see that in individuals that consume higher levels of new 5GC from their diet, there is an increase in the antibody responses, but only against antibodies that recognize new 5GC glycans, but not the human sugar, new 5AC glycans. As a control, we also looked at another sugar, which is called alpha-gal. That is a dietary sugar. It's a galactose connected to underlying sugar galactose. And it's not presented in our bodies, but it is presented on gut microbiome. And all humans have alpha-gal antibodies that maybe you have heard about it in respect to xenotransplantation. And when we look at the antibody responses, we see that there is no change in that, because when galactose is degraded, it is not immunogenic. So that would not change the antibody responses. And we see that the change in the level of antibodies is really related only to new 5GC. Now, when we try to look at the repertoire of these antibodies, looking at the actual sugars that are being recognized, we see a threshold effect when in individuals, in men 45 to 60s that consume higher levels of new 5GC, particularly from red meat, but also from dairy, there is a diversity of response that also the response is higher already in quartile 2, and there is a diversity in the response. And if we look at how the terminal sialic acid is connected to underlying glycans, we see that it's different. You see here at quartile 1, there is more blue, that's the alpha-6 linkage, and in quartile 4, there is more alpha-3. And although it seems very obscure to you, I'm sure you can relate it to something that you know, and perhaps you've heard about the bird flu versus human flu. So bird flu would recognize this type of sugar that is connected to alpha-2,3, while the human flu would bind to the alpha-2,6 linked sialic acid. So we know that the linkage really matters in immunity and also in the diet. Also, the underlying glycans were a bit different. And now we can actually use this linkage to separate the individuals that consume low levels of red meat versus those that consume high levels. So even if an individual would lie, we can now have a measurable parameter to actually see if it's correct or not. And we saw the same pattern in women. So increased new5GC in the diet leads to increased levels of anti-new5GC antibodies with altered repertoire. And now, to really see whether this change in the repertoire is really related only to the diet, we looked at another type of disease, infectious mononucleosis, that is related to EBV infection. And in that disease, there is an overall increase in antibody responses. So we confirmed the increase in anti-New5GC responses and the specificity to New5GC glycans, but not to New5AC glycans. And now when we look at the repertoire, we see that there is no change in the repertoire in patients versus controls, which means that the altered repertoire that we see based on higher red meat consumption is really related only to dietary consumption. And another way to get exposed to New5GC is through drugs. Many of the drugs are monoclonal antibodies that are produced in animal cells, including mouse or rabbits. In this case, we took ATG, it's rabbit anti-thymocyte globulin that is produced in rabbits. Rabbits are mammalian non-humans. They have New5GC, and when the drug is produced in rabbits and given to human patients, they actually get vaccinated unintentionally against New5GC. And when we track the antibody responses, we see that 1 month after this vaccination or drug introduction, there is an increase in the antibody responses. And when we measure the affinity of these antibodies, we see that the affinity of individuals that had been vaccinated have a higher affinity against New5GC. And so we wondered whether that would also be the case in dietary induction of anti-New5GC responses. To do that, we first affinity purified anti-New5GC antibodies from the blood of these individuals, and we see that those that consume higher levels also have the amount of antibodies were higher in those that consume high levels of red meat, and the specificity again confirmed by glycan microarray. The repertoire is clearly different, and now when we look at the affinity, we see that there is no change in the affinity. So if we think about the types of anti-New5GC antibodies, perhaps there's a myriad of possibilities. Some are higher because of our diet, others could be higher because of vaccination. And really to go back to this question of xenosialitis, does it really matter whether you have high affinity or low affinity antibodies? Would that cause disease or mediate a disease or even save you from a certain disease like cancer? So in order to address that, we affinity purified antibodies from drug-induced individuals versus a diet-induced antibodies, and we expose them to endothelial cells, and we look at the gene expression. When we looked at exposure of diet-derived high levels of anti-New5GC antibodies, clearly we see that the gene expression patterns are a mirror image to the drug-induced antibodies. So the high-affinity antibodies actually increased or decreased antibodies that are completely different than the diet-derived. We still are investigating that. We don't know exactly what this means and whether diet-derived antibodies would mediate inflammation while drug-induced antibodies would be beneficial for therapy, but that is still ongoing. To go back to the diet, so I'm often asked, so what does it mean? Does it mean that red meat, we should stop eating it? So the answer is no, and like everything in life, moderation is the key. And what we quantified in order to be in the low quartile of low levels of anti-New5GC antibodies, that would be 1 or 2 steaks per week, while 4 or 5 steaks would put you at risk, at least in developing high levels of anti-New5GC antibodies. And intuitively, to help us actually look at the potential risk, we developed a glycemic index to quantify New5GC in different food items. And compared to steak, if steak is 1, we see that, for example, goat cheese or sheep cheese or goat cheese like camembert has 3 times the amount of New5GC. So if you eat 1/3 of the amount of goat cheese then you would have this high level of red meat, new 5GC in your diet. So this kind of put things into perspective in terms of what is good enough or low enough for our diets. And if we go back to the world population data, if we just separate these numbers into countries that consume below or above this 120 gram of meat per day, we see that if we look at the cancer incidence and cancer mortality, it is dramatically increased in countries that consume high levels of red meat, at least 2.5 or 3 times the risk. So the take-home message is that new 5GC consumption from red meat can dictate the levels of and characteristics of these antibodies, and perhaps could explain the molecular link between red meat diet and cancer risk. And just to finish with another note, we recently published that these antibodies, as I said, cause xenocyalitis and mediate different chronic inflammation-mediated diseases. Cancer is one of them, and recently we've also shown that these antibodies are the culprit for malfunctioning of bioprosthetic heart valve that get calcified and deteriorate in patients after introduction. So with that, I'll thank my group and my collaborators and funding. Thank you very much.
So to put it short and staying with the French, la dépression. Now I'll never look again the same on the piece of a brie, camembert, or red porterhouse. Dino is with me on that, right?
It's better with tequila. With tequila, it solves everything.
Any questions beyond diet recommendations? Yes.
Well, the gene will be there, CMAH. So even if you propagate a tissue, a derived food tech steak from the cells of a native animal, mammalian cell that has a new 5G, CMAH, then you would still have that risk. Yes, one of the things you can do is actually genetically modify, and actually in the study for the bioprosthetic heart valves, we generated new 5GC knockout cows. So we have safe steaks. There is a patent on that, and it's marketed actually already. So yeah, that would be the solution.
Does it taste the same?
I haven't tasted.
A question? No, thank you. Wow, what a day. So to summarize this fantastic day, I would like just to thank first and foremost Dr. Judith Ben-Porat for being my right arm. And of course, Ayelet Rashti for helping out with everything possible, including parking. I would like to thank Jack and Victor for doing an amazing job and broadcasting this live. And this is recorded, so it will remain on YouTube. And all our listeners that are online throughout this day, I would like to thank all the wonderful, wonderful speakers who were just mind-blowing with all the great ideas. I think we will all learned a lot, so thank you, everyone. And of course, thank you to all, and we'll meet here next year. Thank you.